Hello, everyone. I'm Mark McClellan. I'm the director of the Duke Margolis Center for Health Policy, and I'd like to welcome you back to day 2 of our workshop on translating science and drug development, surrogate endpoints, biomarkers, and more. We had a productive day yesterday. We talked both about the current opportunities and the challenges to biomarker development, including the fact that this can be a resource-intensive process, but one where we know how to make it work better. It can benefit from good roadmaps and examples, and from strong collaboration starting early in the translational development process. There are a number of public-private partnerships that we heard from, including hearing about the FNIH, Critical Path Institute, and other efforts that are playing an important role in bringing together parties in a pre-competitive space. We heard about how this is an iterative process that benefits from ongoing discussion and learning and feedback from all stakeholders, including regulators, along the development spectrum. We heard about how translational science can help remove some of the barriers between pre-discovery, clinical development, and regulatory approval as part of this process. We also heard about better predictive models and how they can lead to more precise patient selection and reduce clinical trial risk, with better explanations of variable treatment responses across different patient groups. These kinds of concrete examples and success stories have been very helpful for driving further development. But we also heard about examples where the approaches didn't work so well, and those failures are also incredibly helpful. So today, we're going to build on the sessions that we heard about yesterday, all of those specific examples, all of those topics, and we've got 3 sessions to do that. In session 3, we'll start with a discussion of the validation process for surrogate endpoints. Then session 4 will turn to other translational approaches beyond the use of surrogate endpoints. And then finally, in session 5, our panelists will reflect on the previous discussions over yesterday and today to look forward around the common challenges and the further opportunities to address them. So next on the slide is a short note about our commitment to academic independence and avoiding taking partisan positions in our work. You can find more information about our policies at the link on this slide. I am also an independent director at Alignment Healthcare, Cigna, and Johnson & Johnson. Again, our policies on independence, disclosure are all described at the sites noted here. A short note also that Duke Margolis is convening today's meeting in cooperation with the FDA. This is not a federal advisory committee meeting. We are very interested in input from everyone, all of our speakers and all of our attendees on this topic, but this is not a formal consensus-seeking process. We're not following advisory committee procedures. We're not taking a vote at the end of the meeting and so on. Instead, this meeting will be a success if there is an open, frank, and lively exchange of good ideas on the very important topics that we're covering today. Throughout the workshop, all of you will be able to share thoughts and submit questions over Zoom. Our team is going to review those questions as they come in. We'll route them to session moderators to try to get them raised in the sessions themselves. And as we did yesterday, we'll also try to answer as many of these questions live as possible. So please do contribute. We'll try to get to as many of them as possible. It adds a lot of value to the achieving the goals of this meeting. We're going to have a recording of the meeting as well as the slide deck available on the Duke Margolis event website and the YouTube channel for Duke Margolis after the conclusion of this event. And again, we are very pleased to have so many of you joining us today on these important topics, and we're looking forward to another day of interesting and productive state-of-the-art discussions. With that, I'd like to turn over this session to Norman Stockbridge. Norm is the director of the Division of Cardiology and Nephrology in the Office of Cardiology, Hematology, Endocrinology, and Nephrology in the Center for Drug Evaluation and Research at FDA. And Norm's going to be moderating session 3. Norm, thanks so much for joining us today.
Good morning, good afternoon, and good evening, depending on where you are. I'm Norman Stockbridge. As you've heard, this session is going to be about validation and regulatory acceptance of biomarkers as surrogate endpoints, the holy grail for biomarkers. The first speaker, we're going to have 4 speakers. The first speaker is Dr. Steve Ryder, Chief Medical Officer for Rally Bio. And he's going to be discussing a case study on tools for rare diseases. Steve?
Norman, thank you. It's a pleasure to be invited to this symposium. For the last 8 years, 5 at Alexion and now 3 at Rally Bio, rare disease therapeutic development has been my entire focus. At Rally, this is very much a daily focus. It's what I do. Today, I'll share with you a case example of an assessment tool successfully imported into a rare disease development program. It was introduced to me by David Thompson, Tino Melli, and Kenji Fujita and their colleagues. Next slide. All were at Alexion at the time of this work. Next slide. Rare, ultra-rare diseases are too often poorly understood and researched. This extends both to the preclinical and clinical areas and covers issues of study design, assessment tools, dose, and benefit risk determination. This presentation will provide a case example of where a clinical assessment tool was imported, modified, validated, and successfully used in the Strensiq hypophosphatasia development program. Next slide. Hypophosphatasia is a rare, serious, and potentially fatal genetic disorder caused by loss of function mutations in the gene encoding tissue nonspecific alkaline phosphatase, leading to elevations in extracellular inorganic pyrophosphate. This inhibits bone mineralization by blocking hydroxyapatite crystal formation. HPP is characterized by defective bone mineralization and impaired phosphate and calcium regulation, leading to bone deformity and destruction, pain, muscle weakness, respiratory failure, and gait impairments. Next slide. Asfatase alpha, or Strensiq, is a human recombinant fusion protein developed as a treatment for HPP. The activity of asfatase was established in alkaline phosphatase knockout mice, shown on the right graphic, with a near return to normal life expectancy. Next slide. Clinical efficacy was established through a series of interdependent endpoints beginning at the level of biochemical substrates, extending to measurements of bone mineralization and radiologically assessed pathology, and extending then into clinical assessments of physical function, quality of living, and survival. The irreversible morbidity and mortality of HPP precluded placebo-controlled trials of meaningful duration in children with perinatal or infantile onset and juvenile onset disease. This required an informative and relevant historical control reference. Next slide. For perinatal and infantile onset disease, where the disease is most severe, informative and relevant historical control reference data were available, since the informative endpoints of ventilatory support and mortality are routinely captured in medical records. Next slide. The top panel shows the ventilatory status and patient outcome for 20 matched historical control patients. Nearly all of these patients died in childhood. In comparison, the lower panel shows the ventilatory support status for 14 asfatase-treated patients requiring ventilatory support at baseline. Half of the patients became ventilator-free, and 3 of the 14 patients died, a stark contrast to the upper panel. Next slide. Overall, among patients with perinatal and infantile onset HPP, asfatase patients had a significantly better survival rate compared to historical controls. Next slide. For juvenile onset patients, where mortality is not generally increased, although the morbidity is very real, the assessment endpoints of ambulation, development milestones, and activities of daily living were not routinely captured in medical records, and this posed a challenge. While improvement from baseline was seen in all these measurements, the comparison to an informative and relevant historical control reference was missing. Next slide. The 6-minute walk test was conducted in HPP patients 5 to 12 years of age, and aspartase treatment was associated with rapid and sustained gains in ambulation, entering the normal range after 6 months. Next slide. The BOT2 is used to assess strength and physical function in patients 4 to 21 years of age. Using the same display as for the 6-minute walk test, results show median scores reaching the normal range by 1 year of treatment. The next slide will bring this assessment tool to life, displaying from left to right a patient taking the BOT2 at baseline and after 6 months and 3 years of aspartase treatment. Next slide. So as you saw in those videos, there was marked improvement from left to right. As impressive as the changes in the 6-minute walk and the BOT2 are, comparisons were limited to changes from baseline since these were not included in the historical record. David, Tino, and Kenji noted that at Shriners Hospital in St. Louis, historical video recordings were made of nearly all HPP children extending back for decades. This was a center of excellence in the United States. Video records of the children walking the corridor of the clinic. The concept arose whether these video records could be assessed, matched to the aspartase patients, and a comparison made. Next slide. The literature was surveyed and an assessment of gait was found to be in place. Next slide. The Performance-Oriented Mobility Assessment, or POMA, was introduced by Dr. Mary Tinetti in the mid-1980s as a mobility assessment in the elderly. Dr. Tinetti was at Yale, and the team was actually able to directly consult with her, being New Haven-based. One component of the POMA is an assessment of gait, specifically assessing components such as step length, height, symmetry, continuity, and walking stance. Next slide. Working with expert consultants led by Dawn Phillips at the University of North Carolina, the original scale was reviewed, adapted, validated, and then applied to the HPP trial patients and matched controls. Next slide. Carefully documenting each review and adaptation step, the original scale was modified to focus on those aspects of gait most affected by HPP, preparing the modified, or M, POMA-G, with a 0 to 12 scale of disability. The modifications are listed here and included items such as removing gait initiation, since children don't generally have trouble initiating gait, and removing the rating of path directions, since children may or may not walk in a straight line. Next slide. The modified scale was then validated by correlating the M-POMA-G score with changes on a number of already validated instruments, such as the Childhood Health Assessment Questionnaire Disability Index, CHAC-D, and the Pediatric Outcomes Data Collection Instrument, POD-C, and the 6-minute walk test. The highly significant correlation with the CHAC Disability Index is shown in the lower panel. Next slide. Following validation, the M-POMA-G was applied to video records of 6 historical matched patients and 8 patients receiving aspartase. There were a couple of logistical issues, since As you can imagine, they had to obtain written and informed consent from all of these patients, but it was obtained. Next slide. The median rate of change per year significantly differed between the asfatase treated and the historical control patients, providing support for asfatase in the treatment of patients with juvenile onset HPP. As shown in the bottom, in comparison to the stability of the 6 historical matched patients in the top panel, 7 of the 8 patients receiving asfatase improved, as shown in the bottom panel. Next slide. This work provides a case example of how rare and ultra-rare drug development can be assisted by the logical importation of relevant tools, including their review, modification, validation, and application. Thank you for your attention. Norman, thank you for the opportunity to present.
Great, thank you for that. One quick follow-up question for you before we go to the second talk. You know, some of us probably would have thought that this instrument was a measure of how patients feel, function, or survive. That is, not a surrogate. Were you told that this was a surrogate and that it needed to relate to something else?
No, the interactions with the agency, and I remember the medical officer, Dr. Carla Epps, at the time, were very supportive, and I think that there was full agreement that what was being measured was clinically relevant. So no, it wasn't described as a surrogate, Norman. But I think that there was great excitement about the concept of going back, finding a clinical assessment measurement that could be objectively applied. The videos could be looked at in a blinded manner by 3 raters, and then actual quantitative assessment of the improvement provided. I hope that answers the question.
Great, it does. Thank you for that clarification. Our second speaker is Dr. Henrik Zetterberg, who's professor of neurochemistry at the University of Gothenburg and University College London. He's going to be presenting on a cerebrospinal spinal fluid biomarker for neurodegenerative diseases.
Thank you very much for having me. I'm really delighted to be in this session. It's such an exciting field, and I am specialized in neurochemistry, clinical neurochemistry, working with fluid-based biomarkers for neurodegenerative diseases. And here we have seen a lot of developments recently, which also involves actually recent FDA clearance of some of the assays to measure CSF biomarkers. So we could take the next slide, please. So this is a little bit of a busy slide, but it depicts a neuron in the brain, and the typical Alzheimer's disease-related brain changes are amyloid plaques that you see in the lower right of this picture, and then also neurofibrillary rectangles that you see to the left in the picture in the neural soma. And these 2 key pathologies are present in the brain when you get Alzheimer's disease, perhaps 15 to 20 years before onset of the dementia phase of the disease. Especially beta amyloid plaques are very early when it comes to the onset of accumulation of these plaques. It often starts in the hippocampi and entorhinal cortex, and then the disease spreads across the brain. And it's often regarded as when you enter the clinical phase of the disease, the brain is, you have a lot of amyloid pathology everywhere, but then you also have neurofibrillary rectangles composed of an axonal protein called tau. And the distribution of these tangles correlates with what types of brain regions that are affected, and that also correlates, of course, with the symptoms. So we could take the next slide. Here I just wanted to tell you that this presentation will deal with biomarkers for amyloid plaques and for tau tangle pathology in the form of phosphorylated tau. Next slide, please. We could take next slide also. So if we look into the literature in regards to fluid biomarkers for amyloid pathology of Alzheimer's disease, we have known for many, many years that cerebrospinal fluid concentration of 42 amino acid long beta amyloid is decreased in Alzheimer's disease. And when we develop biomarkers, of course, it's important to know what the biomarker reflects. And we have known for a long time that the core of the amyloid plaques are rich in the 42 amino acid long form of beta amyloid. This is a form of amyloid that all of us make. It is hydrophobic and it can aggregate at high concentrations, and then it starts to form these types of plaques. And this is a templated misfolding reaction, not dissimilar to prion diseases like Creutzfeldt-Jakob disease. So in Alzheimer's disease, lumbar CSF, low levels of this protein, this small protein, indicates that it is stuck in the brain. And young people all have high levels, meaning that they maintain a beta 42 in solution. This has been seen in many studies. This is an extraction of the ALTS biomarker database that is available through ALTS Forum, open access database on the performance of different biomarkers for Alzheimer's disease. Next slide, please. Here we see an old study and the correlation between the biomarker in the body fluid on the y-axis and post-mortem data on amyloid plaque counts. And you see this type of relatively strong correlation between high plaque counts and low CSF beta 42 concentrations. And in the validation phase, it has been important to replicate this, of course, and also to relate it to imaging evidence of amyloid pathology in the brain. Next slide, please. Let's see, there we go. Perfect. And now we have the possibility of measuring amyloid in the brain in living people by amyloid PET, which is also now an approved test for amyloid pathology. And if we click once more on this slide, then we see here the relationship between amyloid PET signal on the x-axis and cerebrospinal fluid A beta 42 concentration on the y-axis. And then you see this type of almost bimodal correlations. You have people who are concordantly normal for both biomarkers. They have high CSF beta 42 concentration and low amyloid PET signal for amyloid pathology in the brain. And then you have people who are in the lower right quadrant who are concordantly abnormal with high amyloid PET signal and low CSF beta 42 concentration. We could click once more and once more. In this study, we also made a replication here to set the cut point, and we see that this is a quite replicable finding. And this overall result has been replicated across many, many studies now from research groups all over the world. And there are actually almost, I think it's 60 or 70 studies that show this type of pattern. So we think now that we understand CSF beta 42 as a biomarker. Next slide, please. But there are problems still, because if we measure CSF beta 42 concentration in people who have neuroinflammation, then we learned in the C and D panels here that patients with active neuroinflammation in the form of multiple sclerosis, they have lower levels of CSF beta 42. But in the A and B panels in the upper part of the figure, you also can see that other A beta forms that are more water soluble, C-terminally truncated A beta 38 and 40 that are in solution and do not aggregate in the brain, they are also lower. And this is not the case in Alzheimer's disease with plaque pathology, because then you have a selective reduction in A beta 42 concentration and the other A beta forms stay normal. Next slide, please. This is the case in normal pressure hydrocephalus also. So if you click once more, you see here that A beta peptides and soluble APP, the mother protein of A beta, all are reduced in normal pressure hydrocephalus. So here, for many years, we had a misconception in the field that normal pressure hydrocephalus had a high prevalence of amyloid pathology, but we now know that this is due to general disturbance in the amyloid metabolism. Next slide, please. The solution to this problem is to make a ratio, CSF beta 42 to 40 ratio. And this is our plaque pathology marker that has now also been approved as an IVD diagnostics. Next slide, please. Here you see the data on what happens to the biomarker performance when you combine A-beta-42 with a water-soluble shorter A-beta form and make a ratio. So in the A panel, the red line indicates the AUC, the diagnostic performance, one could say, of A-beta-42 alone, and you get an AUC of 0.9, which is a good biomarker. But if you combine it with A-beta-40 and 38 in the ratio, you reach almost complete concordance with amyloid PET. I should say here that the reference standard here is amyloid PET positivity, amyloid PET to determine amyloid positivity. These are two different assays, and that has also been an important part of validating this biomarker. Different assays that use similar or the same or slightly different antibodies give the same result overall, and they correlate, and there is also now a reference material and a reference method available to standardize these assays. Next slide, please. The biomarker has, the ratio has a bimodal distribution, which is a fascinating feature of this biomarker. So you are either normal, and that means that you are not depositing, which is the green normally distributed population here, or you are abnormal and you deposit amyloid. And there is a very small gray zone, which is an unusual feature of this test as a clinical chemistry test. Next slide, please. Using the test, we can also follow people when they start to develop amyloid pathology. And this is a study we did together with the Madison researchers, Sterling Johnson and Toby Betthauser. They invited people who were adults but had one or two parents with sporadic Alzheimer's disease, and they gave CSF and underwent repeated amyloid PET imaging. These people are at increased genetic risk for Alzheimer's disease, but it's the sporadic form. And you see here that people, when they are 40 to 50 years of age, if you look at the x-axis, you see that all are normal in the ratio. But from 50 years of age, people start to die down in the ratio, and that means that they start to accumulate amyloid. And that is evident in the rightmost panel here, where you see the trajectories of the amyloid PET signal increase over time. So the fluid biomarker is almost like a qualitative marker of when onset of amyloid accumulation starts. And with amyloid PET, you can quantify the progression of the amyloid accumulation over the brain tissue. And then you also see this interesting feature of amyloid PET, that people have relatively parallel trajectories of the slope at which they accumulate amyloid. Not trajectories of the slope, but the slope is quite parallel, which means that when the amyloid cascade has taken off, it happens independently on how you live your life. And the best way of stopping an amyloid cascade is, of course, to remove the substrate of the aggregation process. And this is also obviously what we all hope that the anti-amyloid antibodies will do, and thereby slowing down the whole process. Next slide, please. One can line up people and define a time point 0 when they start to accumulate. And then you see that a few years later, a few years after people have become amyloid positive according to the CSF test, then they also become amyloid PET positive. Next slide, please. Plasma A-beta is a very hot topic. The blood test for Alzheimer's disease, it has been claimed if we could measure A-beta, but over many years, we have been able to measure plasma beta, but we haven't had any concordant or interpretable results. So 1 in this graph indicates no change. And throughout the years, we have had studies publishing increased or decreased levels, and most published no change. Next slide, please. But now we have an amyloid test in blood, which was developed by Randy Bateman's team in St. Louis, and you will also see some other parallel developments, where in this test you use antibodies to extract all beta amyloid from the plasma sample. You then digest the amyloid and measure the beta 42/40 ratio by mass spectrometry, and then you see this type of reduction in the ratio. We see that we have a clear group level difference and an AUC which is quite high of this test. Next slide, please. We see also here, you can push once more or click once more. This is another type of assay from an Australian-Japanese collaboration, also mass spec based, where you pull out A-beta with antibodies, and then they actually here used an inverted ratio, which is the reason for why you see increased and not decreased levels of A-beta in amyloid positive people. Next slide, please. We could actually skip this slide and jump to, if we click once more, we can click once more. There we go. The mass spec-based assays, they are difficult to run in a clinical laboratory setting on samples that come in ad hoc. So we have worked together with Roche to develop an immunoassay. And this is data from an assay we developed on the Cobas platform. And in the left part, you see amyloid negative people, and in the right part, amyloid positive people. And there you see you get by this clinical chemistry fully automated test is around 0.8 or 84 in this case. But you also see that individual patients end up in, it's very hard to tell if an individual patient is amyloid positive or negative. Next slide, please. And here you also see part of the problem. In the B panel, you see the fold change of the A-beta ratio in CSF with a 50% reduction and a small overlap of patients who are amyloid PET positive and negative. And then in the leftmost A panel, you see that the reduction in plasma is only 10, 12% because it happens on top of peripherally produced amyloid. And this is really the challenge with this amyloid test. Next slide, please. We can jump on more. And this is another way of putting it. If you look at the rightmost part of this figure, you see CSF phospho-tau A-beta 42 ratios. And you see that PET negative people who don't have amyloid, they end up nicely in the lower right of this graph. And amyloid PET positive red circles, they are in the leftmost part. And there is a lot of wiggle room in the C panel where the assay could drift a little bit without causing too many people to be misclassified. But if you look at the plasma A-beta 42 ratio in the leftmost part of the panel, you see this group level difference, but you see that amyloid PET positive and negative people are very close. So if anything happens to this test in clinical laboratory practice, you will have a lot of people moving across the threshold. So this is a little bit of a scary assay to use in a clinical chemistry lab week by week, because it has in clinical chemistry we would call this test not robust enough, or sometimes people, clinical chemists also talk about the ruggedness of the test. Next slide, please. So if I would ask the question, can we diagnose amyloid pathology with a blood test? Are we there yet? For group level enrichment and screening, I would say yes, if you accept that individuals might be a little bit misclassified. For example, as a pre-screen for entering a clinical trial where amyloid pathology will be confirmed by CSF or PET. For individual diagnostics, we are not there yet, but maybe one can at least say that we start to see some results that are starting to look really promising. Next slide, please. I will now end with tau pathology, and I will be quite brief here also. So next slide. Phospho-tau is increased in Alzheimer's disease. That is known for a long time, and that comes from neurons that are exposed to amyloid, and they phosphorylate and secrete tau, and the levels in the CSF get higher. Next slide. But in other tauopathies like frontotemporal dementia and PSP, the levels are low. So this is an Alzheimer's specific test, and it reflects the amyloid, the disturbance in tau homeostasis in amyloid exposed neurons. That has been shown by a lot of basic science research in iPS-derived neurons, animals, and also in patients where it has been possible to measure A-beta production and clearance rate by a certain methodology called SILC. Next slide, please. We could click once more. There are different forms of tau that we can measure, different forms of phosphorylated tau, but they behave relatively similarly. Next slide, please. And we can now measure this in blood. And in the leftmost part, you see the change of phospho-tau levels to 31 in patients with super small amounts of amyloid in the brain, and then increasing amounts. And you see that already when patients get a little bit amyloid positive, the plasma biomarker detects that. And then it increases to 3-fold, even 4-fold in blood. Next slide, please. It can be used in diagnostic algorithms to predict Alzheimer's disease dementia in people with subjective or mild cognitive impairment. And if one only does the plasma phospho-tau test, there you see is 0.8 in this study. If you also include some brief cognitive tests, you reach 0.9. Next slide, please. You can also use different types of phospho-taus and reach the same conclusion of around 0.9 in AUC. Next slide, please. And this reacts also to treatment. So this is donanemab, an anti-amyloid antibody, lowering plasma phospho-tau217 with about 10%. Next slide, please. The controversial aducanumab drug lowers plasma phospho-tau181 with 13 to 16% in this study. Next slide, please. So if one would conclude on tau, we now have a test that works both for group level enrichments and screening, but also for individual diagnostics. It's getting there, and it really looks like this test. I think we will see tests that have become clinically validated and up and running, at least in Europe, in clinical chemistry routine during this year, actually. Next slide, please. With that, I would like to conclude and just mention then that the work on these markers have been, it has taken many years to validate them, and it has been very important to link the biomarker results to the brain pathologies they are meant to reflect. And this year we will see a lot of different studies examining these biomarkers in more diverse populations with comorbidities, and a lot of those things we need to figure out when starting to use these tests in clinical chemistry practice. Thank you.
Yeah, thanks very much for that. Can you answer one quick question for me?
Yep.
You know, for the former biomarker, it's at least a little unusual to try to validate it against another biomarker. Yep. Would you want to comment on that?
It has meant a lot to this field to be able to use amyloid PET as the reference test for amyloid in the brain. And that has been possible because amyloid PET was validated against neuropathology and approved for that. So that's sort of the traceability chain of the validation process. But it is, of course, important to also make sure that this is correct and possible. So there are approximately 5 or 6 studies that have done a direct validation of CSF tests for amyloid pathology to neuropathology. And I think there are a couple of tests also now on the plasma beta 4240 test.
Okay, thanks for that. Our third speaker is Dr. Leslie Inker. She is a professor of medicine at Tufts University, and she is going to be discussing GFR decline as a surrogate for end-stage kidney disease. Leslie?
Thank you. Thank you so much for the invitation to speak to you all today. As mentioned, I'll be speaking on GFR decline as a surrogate endpoint for progression of chronic kidney disease. Next slide, please. So kidney disease is a very slowly progressive disease. Clinical trials to evaluate treatments to prevent or slow the progression to kidney failure require long duration of follow-up, leading to expensive and complex trials or highly selective subset of participants. Oh, excuse me for the typo, but doubling of serum creatinine, which is equivalent to basically a halving of GFR, is accepted by regulators, but still occurs late in the disease course. And these challenges have likely contributed to the paucity of therapies to treat kidney disease, as seen on the figure on the right, which shows on the bottom 2 lines the amount of trials in kidney disease relative to many other domains in the medical field. Next slide, please. In the realm of chronic kidney disease, we have 2 central biomarkers, GFR and albuminuria, or more generally proteinuria. And these have been part of our definition for chronic kidney disease and part of the staging system as per the KDIGO guidelines, international guidelines for chronic kidney disease, already published a decade ago. Next slide, please. So as part of our research group, the Chronic Kidney Disease Epidemiology Collaboration, we have done investigation of surrogate endpoints for trials in CKD progression for now almost 2 decades. It started with the NIH and the NIDDK asking us to include evaluation of urine protein as a surrogate when in our grant that actually looked at something completely different, which is estimation of GFR. But since then, we've been very fortunate to have sponsorship from the National Kidney Foundation and have done our work in collaboration with the FDA and then more recently with EMA to evaluate both changes in urine protein or albuminuria as well as GFR decline over the years. Much of that work had been done for a series of conferences addressing a particular question. And then following our very successful March 2018 2018 workshop, we said we actually need a more stable funding structure to allow us to address the continued methodological questions and to incorporate new studies that are now in existence in kidney disease. And so again, with support of the National Kidney Foundation, we've raised funds from sponsors for now 2+ years of funding to have enhanced method development. Next slide, please. So let's now turn to looking at declines in GFR specifically. What I have here is a very hypothetical figure of the time course of kidney disease. And actually 14 years might even be considered fast for CKD progression from the onset to development of kidney failure, as you see that I've drawn here at 14 years. I've also shown on here doubling of creatinine, which, as I mentioned, is a 57% GFR decline, and it still occurs many years after the onset of kidney failure. Next click, please. So in March, for the purposes of a workshop with the FDA, we looked at lesser declines in GFR and specifically looked at 30% GFR decline and 40% GFR decline. And these were published in a series of papers that I've outlined here. And again, these are actually fairly late in the disease course. Next click, please. And so for the March 2018 conference, we then turned to look at slope. And these are outlining some of the publications I listed here, but that's what I'm going to be talking about for the rest of the presentation. Next slide, please. So what are some advantages or limitations of slope? Why did it take us so long to get to look at this specifically? Well, the advantages of it is that regardless of the cause of kidney disease, decreased GFR defines chronic kidney disease. The level of GFR indicates the severity of chronic kidney disease, and GFR decline is the definition of progression for all causes. And compared to the time to event endpoints, there's increased power because you can use all participants. It includes both fast and slow progressors in the assessment of the outcome, and includes patients who have GFR decline that might lead to the endpoint even after that it will occur after the ending of the trial. But there's some limitations and complications. The estimated GFR, which is most often the measure used, not the measured GFR. But estimated GFR can reflect the true GFR as well as non-GFR determinants of the serum creatinine level or whatever filtration marker that's used. There's also concerns about nonlinearity, heterogeneity in the overall group assessment of decline in GFR over time. Issues of informative censoring, that patients who started dialysis or who died can't contribute to the slope, but that's informative to that their loss of information informs the changes between the treatment groups. And of course, and most importantly, is the fact that acute effects or immediate effects from the intervention differ from the long-term chronic effect of the treatment. Next slide, please. And this acute effect in particular has really been a challenge with all of the studies in chronic kidney disease. So let's describe what that is. Here on the x-axis, we have time, and on the y-axis, we have level of GFR. In the green is the control arm, and in the blue is the treatment arm. So in the control arm, you assume we're over the overall group, there's a linear decline in GFR over time. In the blue arm, in the intervention arm, it's different, where the intervention, in this hypothetical situation, we have the intervention which causes an immediate decline in GFR compared to the control slope. But after that, there's a stabling of the effect. And there's subsequent, the slope after that, what we call the chronic slope, has a shallower slope compared to the control arm. And this is because if we think of what we're measuring or estimating is the overall concept of GFR, but that's a component of the number of nephrons and as well as the work of each individual nephron or the single nephron GFR. And the hypothesis that in the control arm, there's a declining number of nephrons because of the cause of kidney disease, calling nephron loss, but there's actually a stable work of each individual nephron, a stable single nephron GFR. The treatment arm, in contrast, there's a short-term change on the single nephron GFR, decreased filtration, as well as this long-term stabilization of loss of nephrons. And in fact, the predominant hypothesis in chronic kidney disease for now many years is that the that the actual decline in the loss of hyperfiltration on the work of individual nephrons is part of the reason why we have stabilization of nephron loss over time. And so when we needed to incorporate consideration of this essentially two-phase component of the GFR slope when we analyzed it for consideration of surrogacy. Next slide, please. So how did we do this? Well, our goal was to provide a set of models that accommodated the range of circumstances that were expected in trials of CKD progression. How did we deal with the concept of a linearity or the challenge of linearity? In general, what we found is there's a reasonable assumption that moderate deviations from linearity in the chronic phase do not affect the overall slope estimates in trials that have a relatively short in duration. And we had, therefore, a two-slope model to accommodate acute treatment effect in GFR that differs from the chronic slope. Next slide. We included in our shared parameter model informative censoring for studies with greater than 15 kidney failure or death events. We used a shared parameter model with reliable survival times. We accommodated heterogeneity using between subjects with random slopes and intercepts within subject, a power of the mean model to allow greater variability at higher levels of GFR, and heterogeneity in treatment effect. We accommodated by allowing different slope variants in each group to accommodate non-uniform medical treatment effects. So an altogether extremely complicated model that we were very grateful for a senior biostatistician who's been thinking about kidney disease and slopes for a long time. Ed Bonish led our work in developing this model, and this is a paper that's published in Stats in Medicine. So the question is, how could these models apply to each of the individual studies we had? And for the analysis here, we had 47 different trials. And so we developed an automated algorithm that would select the most complicated model, followed by models that did not have one or more of their parameters. And so over all of the 47 studies, we're able to develop the best slope model for each study. Next slide, please. In a trial, so then the question is, how does the treatment effect on the surrogate, the clinical, the GFR slope compare to the treatment effect on the clinical endpoint? And so we did an individual patient meta-regression. We had individual patient in all of these 47 studies. And this was important because we allowed to have consistent definitions for the endpoints and for the slope. And very importantly, we could also account for the correlation between the errors and the estimated treatment effects, so that we weren't overcalling an association on the treatment effects of the two endpoints. We first did a within-study analysis looking at the treatment effects on the GFR slope, which is simply a mean difference between the treatment arms. And then we did the estimated treatment effect in the clinical endpoint using a Cox model expressed as the hazard ratio. Next slide. Oh, sorry, go back. And then we used a Bayesian meta-regression to obtain the estimate of the regression line between the two treatment effects, as summarized by the slope, the intercept, and looking at an overall fit of root mean square error and R squared. And then we also added prediction intervals for the hazard ratio on the clinical endpoint. So that allows us to say for a future trial, what would the estimated treatment effect be given treatment effect on the GFR slope that will be observed. Next slide. So here's the main result. On the x-axis, you have the treatment effect on the GFR slope, which is the mean difference. And on the y-axis is the treatment effect on the clinical endpoint, which is the hazard ratio. I don't think you can see my mouse, so I'll try and describe in words. On the left, we have the total slope that was estimated over 3 years. So from the baseline all the way to 3 years. And on the right side, we have the chronic slope. This is just at the period after the acute effect phase, which would be after 3 months. Each of the dots represents a trial, and the size of the trial, the size of the circle represents the confidence that we have in the estimate. So the big circles are basically big trials with a lot of events. On the bottom right-hand quadrant, you can see agreement that there's a benefit on the GFR slope and a benefit on the clinical endpoint. And you can see overall, there's a lot of agreement. Most of the trials are in the bottom right-hand quadrant or their top left-hand quadrant. And the overall R squared, which describes the strength of the association, was 0.97 or 97% for the total slope and 96% for the chronic slope. So overall, extremely strong agreement for both of these slope models. Next slide, please. So then we said, okay, how can we then inform future trials? How can we take this regression between the observed past trials to help us inform future trials? So we had looked at, as I showed you in the previous slide, we did a trial level analysis in previous RCTs, and this characterizes the causal association between the intention to treat based estimate of treatment effects and surrogate endpoints. We took that and then we applied it to a new RCT, and we converted it to a probability of clinical benefit for a newly tested intervention. And so that's what you have this, the table on the bottom right-hand side. So let's say you had a estimated effect on the GFR slope of 1, and this is really small, a 1 mil per minute difference in 1 mil per difference per year between the treatment and control arm. So if I had that observation and I had a large RCT, I would predict that my median hazard ratio and 97% prediction interval would be 0.62, and I'd have a positive predictive value of greater than 99% for a clinical benefit of a hazard ratio of less than 1. If I had a smaller difference between the treatment arms of a 0.5, which is still really small, it's still a highly probable clinical benefit with the estimated hazard ratio of 0.77 with confidence intervals that are less than 1. Here you see 0.59 to 0.99 with a very strong positive predictive value for that clinical benefit. If I have a smaller trial, I have a lower probability of lower confidence in that probability with confidence intervals that overlap 1. Next slide, please. We then, under the direction of Tom Green, another statistician who's leads this initiative with me, we said, okay, there's many more parameters that are going to happen in a future trial. How can we try and provide information to those designing trials to accommodate all of these different parameters? And so he did development of simulations to look at based on time, GFR, and rate of progression, and compared the potential benefit using different endpoints. As you can see here, there's generally benefit, the chronic slope and the total slope are in the orange and red, their symbols are lower compared to the other ways showing benefit. So there's relative gains in power for slope analysis when the baseline GFR is higher. Next click. But this slide looks a lot less clear, and that's because we've added in the consideration of acute effect. And this is really a critical consideration selection of total versus chronic slope versus other endpoints. Next slide, please. So what endpoint can I use for a trial? Hita Lampers-Hirschberg and I tried to summarize all of the considerations that is an IMPRESS paper in AJKD. Next slide. And it's complicated, and we do need actually sponsors and other investigators to think about it. I've just put here, blown up some of the previous slide, but there are issues of population issue, considerations of acute effect, as I've emphasized, and other considerations. And all of these would go into the decisions as to which of the endpoints should be used. Surrogate, clinical endpoint, which surrogate. Next slide. Our next steps in our current work, as I mentioned, are to update the set of studies, because there's been some very well-powered big trials in kidney disease, and we wanted to ensure the stability. We're continuing to work on methods work, specifically acute effects or subgroups or interactions, as well as how to combine slope with albumin area, as can be used, especially earlier in phase 2 studies. Next slide. So in summary, we provide empirical data to support use of GFR decline as surrogate endpoints in randomized trials evaluating therapies. When applying these data to design a future trial, the most appropriate endpoint needs to be considered in the context of the trial phase, specific population, treatment, and design. Next slide. And just a huge thank you to our huge team over the many years that have helped us. Particularly want to point out Andy Levy, who was the initial director of CKD-EPI and now is my co-director. Tom, who I've mentioned, who has been working with us since the beginning and has really, we're complete partners in this to try and account for all of the uncertainty, but to provide help to sponsors and investigators to design future trials. Thank you very much for your attention.
Thank you, Leslie, for that presentation. And you know, as you're well aware, we've been following along on this process you've gone through for these many years. And I'll be interested in, and frankly, you've had a lot of influence on at least my thinking about what the appropriate quantitative approach to validation looks like. But I'll be interested in hearing during the panel session your thoughts on what about this development process you think worked efficiently, and what you would advise people to do in the future, you know, in the hope that you know, other projects won't take 20 years to get from, you know, start to finish.
You want me to answer, or waiting for the panel?
Well, I think that's probably not going to be a short answer, so I just wanted you to be thinking about it for when we get into the panel here in a minute. But let's get our fourth speaker up. The fourth presentation is from Dr. Nicole Gormley, who's with the Division of Hematologic Malignancies 2 at FDA. And she's going to be talking about surrogates in oncology. Nicole?
Great, thank you. So good afternoon, everyone. I'm Nicole Gormley. I'm the Division Director of the Division of Heme Malignancies 2 at the FDA. And I was asked to speak with you today about some of the surrogate endpoint considerations in oncology clinical trials. And I'd like to thank the organizers for the invitation to present today. It's really been a very interesting 2 days of presentations and dialogue. Next slide, please. Perfect. So just a brief outline and what I'd like to cover today. So I'll start by sharing some of the regulatory considerations for biomarker development, and then I'll provide a couple of examples with pathologic complete response as one example, and then use of minimal residual disease in multiple myeloma. And then also lastly, just touch on some future directions. Next slide. So there are multiple potential uses for biomarkers, such as minimal residual disease or pathologic complete response. And they're generally recognized as prognostic biomarkers, but there are potential clinical and regulatory uses for these biomarkers. And while there is some overlap, the clinical and regulatory uses often have different purposes, and the trials and data needed to support its use in each of these settings are generally different. Clinical uses include monitoring for or screening for disease detection, monitoring for relapse, guiding clinical decisions, therapeutic decisions, whether that be escalation or de-escalation of care. Regulatory uses include using these biomarkers for stratification, patient selection or enrichment, risk-based treatment assignments, and potentially use as either intermediate or surrogate endpoints. And with these regulatory uses, there's increasing risk such that there's relatively little risk associated with the patient stratification factor if the biomarker is not accurate or robust. But there's significantly more potential risk when it's used as an intermediate endpoint or surrogate endpoint for regulatory drug approval decisions. And we'll go through some of these uses in more detail in the upcoming slides. Next slide, please. So there are three terms that can often are used when referring to a biomarker as an endpoint. The first is an intermediate clinical endpoint. It is a measure of a therapeutic effect that can be measured earlier than morbidity or mortality, but is still reasonably likely to predict clinical benefit. The second is a surrogate endpoint reasonably likely to predict clinical benefit. And then the third term is a surrogate endpoint. And then this final instance The endpoint has been fully clinically validated. The marker has been demonstrated to predict clinical benefit. And we'll go through the process of this clinical validation more in subsequent slides. But the first two endpoints are used to support accelerated approval, while the third, is used to support regular approval. And while I'm describing here the process for establishing the surrogate endpoint, it requires clinical validation. The assay also that's used should be analytically validated. Next slide, please. So there are various considerations when thinking about development of an endpoint for regulatory use. And next slide. Historically, the Prentice criteria has been put forth as a statistical operational criteria to validate potential surrogates. But the Prentice criteria can be summarized as one, the requirement that the surrogate must be a correlate of the true clinical endpoint, and two, that the treatment effect on the surrogate should capture the full effect of treatment on the clinical endpoint. It's essentially the notion that the treatment is irrelevant for predicting the true clinical outcome if you know the surrogate outcome. But it's generally thought that the Prentice criteria are too stringent and not really attainable. And as such, other statistical methods have been developed to validate the proposed candidate surrogate. And one approach frequently used relies on meta-analysis. Next slide. So when considering a meta-analysis for validation of a surrogate, there should be patient-level data from multiple clinical trials. This allows for assessment of the individual level and trial level surrogacy. And when we talk about individual surrogacy, we're referring to the correlation between the candidate surrogate and the true clinical endpoint on an individual level. And trial level surrogacy is correlation between effective treatment on the candidate surrogate and the effective treatment on the true clinical endpoint. But having individual patient level data also allows for determination of the surrogate threshold effect. And this is the minimum treatment effect on the surrogate that's needed to predict an effect on the true clinical benefit, which we'll go through this in detail a little bit more. Next slide. When thinking about using a meta-analysis, there are several considerations in the conduct of a meta-analysis for potential surrogacy evaluation. Ideally, there should be inclusion of more trials, as this increases the statistical rigor of the analysis and may allow for more interrogation of the data to address any remaining uncertainties. There should be inclusion of trials with a wide range of treatment effects, both positive and negative trials, as this will increase the accuracy and then precision of the trial level surrogacy assessment. And then when designing a meta-analysis, consideration of an MRD or specific biomarker, the timing of the assessment is really important. And missing data should be also assessed and accounted for in the analyses. And ultimately, the trial populations and treatments included in the meta-analysis inform the future applicability of the surrogate biomarker, such that there should be adequate information to allow comfortable extrapolation to other settings. Next slide. So I'd like to discuss pathologic complete response and its development as a clinical trial endpoint. Next slide. So this effort was really spearheaded by the collaborative trials in neoadjuvant breast cancer group. And they conducted a pooled analysis of mature trials that had both pathological complete response and long-term outcome data. Next slide. So the objectives of this analysis was to determine the association between pathological complete response and long-term endpoints, such as event-free survival and overall survival. Also to determine the definition of pathological complete response that best correlated with long-term outcomes, as several definitions were used in the clinical field at the time. Also to identify breast cancer subtypes in which pathologic complete response best correlated with long-term outcomes. And then to determine what magnitude of the pathological complete response improvement predicted long-term clinical benefit. Next slide. So ultimately, there were 12 trials included in the analysis, and 3 different definitions of pathological complete response were evaluated in the pooled analysis. And it was determined that absence of residual invasive tumor in both the breast and the axillary lymph nodes was better associated with improved event-free survival and overall survival than the absence of residual invasive disease in the breast alone. Next slide. And the meta-analysis established that at the individual patient level, achievement of pathological complete response was associated with a substantial reduction in risk of death compared to those patients with residual tumor. So firmly establishing that pathological complete response as an individual patient prognostic biomarker. Next slide. But as was stated earlier, trial level surrogacy measures precisely how the effect of the treatment on the true endpoint may be predicted based on the observed treatment effect on the surrogate endpoint. And at the trial level in this meta-analysis, there was relatively little association between pathologic complete response and the treatment's effect on either event-free survival or overall survival. Next slide. So in summary, next slide, there was not, next slide, a pathologic complete response association with long-term outcomes at a trial level, but this was observed on an individual level. So again, indicating that it's a prognostic biomarker. Next slide. A standard definition that includes assessment of the lymph nodes was recommended to be used in future trials based on the results of this meta-analysis. Next slide. And the magnitude of pathologic complete response improvement that predicted long-term clinical benefit could not be established due to multiple factors, possibly due to heterogeneity of the population included in the trials, low attainment of pathologic complete response rates, and lack of targeted therapies. Next slide. So I'd like to switch gears now and talk a little bit about use of minimal residual disease and multiple myeloma in some meta-analyses that have been conducted. Next slide. So Langren and colleagues conducted a trial level meta-analysis, and 4 studies provided data in the evaluation of the relationship between minimal residual disease and progression-free survival. And 2 had information that could be used for the relationship between MRD and overall survival. And overall, the meta-analysis showed that patients who achieved MRD negativity compared to those that remained MRD positive had better progression-free survival with a hazard ratio of 0.35. And those patients who achieved MRD negativity also had a better overall survival. Next slide. An additional meta-analysis was conducted by the Dana-Farber group, Dr. Munshi et al., and found similar results. And in their meta-analysis, they included 14 trials and used the evaluation of MRD and progression-free survival. And then 12 trials were used for the overall survival analysis. And again, MRD negativity was associated with better progression-free survival and overall survival. Next slide. So despite these trial level meta-analyses, next slide, there are many remaining questions surrounding the use of MRD in multiple myeloma. Next slide. The first being, does MRD have trial level surrogacy using individual patient level data? Next slide. What threshold best correlates with clinical benefit? Next slide. What's the appropriate, next slide. What's the appropriate timing of the assessment? And then also, does sustained MRD better correlate with long-term outcomes? Next slide. And then also, should MRD be assessed in only those patients that are in CR or VGPR? Next slide. So I'd like to discuss now the results of the Bellini trial and present it as a somewhat cautionary tale. So again, in multiple myeloma, the Bellini trial, next slide, was a phase 3 double-blind randomized controlled trial of bortezomib and dexamethasone with or without venetoclax in patients with relapsed refractory multiple myeloma who had received 1 to 3 prior lines of therapy. Next slide. So the overall response rate was favorable in this trial with 14% absolute improvement. There was a 12% difference in MRD, and the progression-free survival was also statistically significant with a hazard ratio of 0.3. Next slide. Unfortunately, the investigational venetoclax also demonstrated a worse overall survival in this trial with a hazard ratio of 2.0 observed. Next slide. So when looking at the subgroups, it's notable that patients that had translocation 1114 had a slightly different outcome. And unfortunately, there were only 35 patients in this subgroup. So it limits our ability to draw meaningful conclusions, but nonetheless, in this population, the progression-free survival hazard ratio was 0.1, And although there were too few patients, again, to draw any meaningful conclusions, the overall survival results did not seem to show the same detrimental trend that was observed in the non-selected population. Next slide. So next slide. What are the implications of this? I think it underscores that we need evaluation of endpoints that can be assessed at early time points and late time points that provide definitive evidence of clinical benefit. The Bellini trial, while showing these divergent results, I think, again, just sort of underscores the need for to be able to look at overall survival. And then additional information is needed before reliance on MRD as an endpoint in multiple myeloma. Next slide. Next slide. So I'd like to provide just a few more specific examples of where we are with the use of MRD. MRD has been used to support accelerated approval in ALL. And this was the blinatumomab approval for MRD positive B-cell precursor ALL. In that setting, the approval was based on MRD response rate and hematologic relapse-free survival. Next slide. MRD results have been included in the prescribing information in CLL and both venetoclax and obinutuzumab labels. Next slide. And they've also been included in the prescribing information in multiple myeloma in daratumumab label, Abecma label, but it's currently recommended as a secondary endpoint in oncology clinical trials in hematologic malignancies. The next slide. And then there's ongoing group efforts to evaluate multiple myeloma and use of MRD as a potential surrogate endpoint. Next slide. So in conclusion, validated endpoints are needed for regular approval. Pathologic complete response and MRD are not validated surrogate endpoints, and there's existing uncertainty and remaining questions regarding these endpoints for regulatory purposes. MRD, pathologic complete response, and other biomarker assessments in clinical trials should be discussed with the agency, but the FDA is committed to working with the community on development of these biomarkers to help expedite drug development. Next slide. These are my acknowledgements. Next slide. Thank you very much for your attention.
Great. Thank you for that presentation, Nicole. I did have one question for you too. We often struggle with how to power a confirmatory study and look to a quantitative model of the relationship between the biomarker and the outcome, and then looking at the effect of the treatment on the biomarker that we need to factor in, as well as trying to decide whether or not a confirmatory study is reasonably powered to show a treatment effect. I was interested in this surrogate threshold effect concept. We have sort of modeled this as, what's the uncertainty level in the two relationships we have to deal with. One, the external data that says how related the biomarker is to the outcome, and then what the confidence limits are on the effect of treatment on the biomarker, and we use those things, the confidence limits around both of those things, as a way of figuring out whether or not we think the confirmatory study is adequately powered. Your approach to threshold effect seems to be the same idea. Do I have that right?
Yes, exactly. And I think that's just sort of the strength. If you do have patient level data, you can make sure again that all of the terms progression-free survival or overall survival are defined the same way in these trials, some time points, et cetera. But then that can allow for an analysis to say, what degree of change in MRD, for example, would correlate with, what degree of change in the eventual, outcome of interest, whether that be progression-free survival or overall survival. But it does require, more granular analysis and data to be able to have comfort determining that.
Okay, thanks again. As we move into the panel session next, we're joined by two individuals who didn't have a presentation today. First is Dr. Jeff Allen, President and CEO of Friends of Cancer Research. Dr. Allen, do you want to say some words of introduction?
Sure. Well, thank you for the opportunity to join the discussion today. I think no surprise by the name of my organization, I'm most familiar with the examples that Dr. Gormley shared in her presentation. But I certainly agree with her assessment that these are very informative case studies that hopefully the broader oncology community can learn from and apply into the future. One area that we've been working on related to that is to leverage some of the learnings from both the pathologic complete response example and somewhat from the MRD and multiple myeloma example that she shared, was to, or is currently looking at similar approaches around the potential validation of circulating tumor DNA as a potential intermediate endpoint as well, and have been trying to aggregate the data across numerous different clinical trials in a similar manner to try and leverage data that was being collected already. And so what we were able to identify across a number of different trials, first in a prototype of lung cancer, patients treated with immune checkpoint inhibitors, was to aggregate trials that had similar characteristics in terms of the collection time for plasma samples, and find a common analysis method. Nicole mentioned early on in her presentation around the importance of also characterizing the diagnostic tests that were being used in these instances. And that's very true in the liquid biopsy space, where there is a diversity of different methods and tests that have different characteristics. So trying to align on common measures, of course, is important in looking across multiple different trials. And associating the changes, in this case, with circulating tumor DNA and long-term outcomes. In short, what we were able to see in the lung cancer prototype was that a reduction in circulating tumor DNA following treatment with immune checkpoint inhibitors in advanced lung cancer was that it was associated with improved outcomes in both progression-free survival and overall survival. And we're trying now to try and leverage those methods to further characterize the changes in ctDNA, looking at whether we see similar observations in different tumor types and across different drug classes as well, because biologically things could differ across different tumor types, or timing be different in response to different drugs. But the point being, while I think that hopefully this will be informative to the science, the strategy of having multiple research organizations, whether they be academic research and investigator studies or formal sponsors studies, having common methods that are prospectively defined can help try and standardize some of these approaches to hopefully move the field forward faster and characterize some of these the biological observations in a way that may ultimately be more beneficial than having everyone trying to establish their own individual methods within their isolated studies, as opposed to trying to come up with the best uniform approach across multiple different studies, and actually make progress faster in terms of characterizing whether these endpoints are actually applicable.
I think you've struck a theme that other people will endorse, this idea that there's probably nothing as important as having ready access to the available to the existing data. It's not and having some discipline about aggregating it for the shared use. I think that's a key feature, doing this efficiently. Our last speaker, panelist is Dr. Eliza Thompson. Do you want to say a few words?
Hi, so my name is Eliza Thompson. I am the Deputy Director of the Division of Cardiology and Nephrology. So I work under Norman, who's moderating this session for us. I may make just a few quick comments on Dr. Inker's presentation. So my division oversees many of the products being developed for kidney diseases, and been very fortunate to work with Dr. Inker and her colleagues over the years in terms of tackling some of, really the challenging issues for us. Historically, we had used a doubling of serum creatinine as a surrogate endpoint for progression to kidney failure. And though that worked, if you were talking about people who were progressing at a very rapid rate at sort of the later stages of disease, it was not an optimal endpoint if you wanted to target earlier stages of disease or perhaps populations that aren't progressing as rapidly. And so, over the years, we were able to work with, Dr. Inker, with the National Kidney Foundation, really using this database that they'd established and continue to build upon to really understand how we could, make trials even more efficient, using lesser declines in kidney function and ultimately, slope-based endpoint. I think what I want to highlight is perhaps, Norman, the issue you just highlighted, which is, in these diseases, at least, what we're talking about is actually a common disease, but the ability to aggregate data from a number of different sources, subject level data, is critical. And to do that, I think, Leslie and her group really had to build the trust of the community, right? Both academics as well as industry. I think probably there've been a lot of discussions about data sharing agreements, et cetera. I think the other thing that this speaks to is really getting someone to fund this, right? I mean, you have to create this place that's going to aggregate the data. Unfortunately, I think Dr. Inker's group has been successful in convincing, industry to help support these efforts. The other thing that I think is great about Dr. Inker's group and what they're doing, the CKD-EPI-CT, if I'm saying it correctly, is that, this recognition that it didn't make sense just to have these sort of one-time workshops and then walk away. And then, 3 years later, when we're struggling with an issue, to reconvene. It made much more sense to really have a system that we could always be using to interrogate the data, essentially as each question came up, to be able to go and look at the data. So I think it's really been a real, wonderful example of success.
I think we're already well into question one. Why don't we get the questions up on the screen? And then Leslie, do you want to respond to this this challenge of how you get access to data and enable this kind of work?
Yeah, thank you. It's interesting because it used to be actually easier, where it was based more on trust, and we're fortunate, I think, in the nephrology community to be generally collaborative. And I think Andy Levy, who was our initial director, had a lot of trust with investigators, and that allowed the first data sharing. It's now there's more emphasis on data sharing and the importance of it, and it's a lot harder. I've been working on 2 years of a data agreement, and we almost got it, and then they forgot to add an attachment. So we're still not even finished that. And it's really a struggle. And I understand there's a lot of security issues with Europe with the new regulations, which is at a challenge, but I'm not sure that it is more protective. And I think that there should be ways to help to have more sharing that can be faster. I know there's a lot of organizations that exist to do that, but I don't think they are actually effective at making it much easier, especially when you do want, as we do, the individual data to try and define variables the same. Just for example, age can't be in categories, and so when we talk about GFR, that does lead to variation in it, and that could lead to more error in the issues, in the estimates. So I think trying to respect the privacy and respect the regulations, but still facilitate in ways to provide data would be really helpful. And having that maybe part of even the agreements of the initial steering committees. At times the steering committees want to appropriately really do all the analyses that they should do as part of understanding the total impact of a trial. But I think having still the process of sharing with us and sharing with other groups like us to continue to work and to incorporate it while they're doing those papers, I think would be really helpful. And we've been very fortunate to have the NKF being our champion to try and raise funding. But it is a lot of work. And I think if there was even more understanding and more saying, yes, this is part of the money that we spend to do drug development is to support methodological groups like ours. That could be one a strategy that would help just make that component just a little bit easier.
So which of those things do you think is actually the rate limiting step for you? Has it been funding? Has it been data access?
Well, rate, we would probably work regardless of funding. I shouldn't say that so badly, but we really love this, and it's we're very devoted. So I think without funding, we couldn't do any work. We probably work much harder than we're funded at. So I would have to say data access.
Henrik, do you want to comment on data access issues and perhaps the European perspective on this?
Yeah, I could mention the GDPR is the problem Leslie's talking about here. It is possible to, different European universities have taken different, they are different in how rigorously they implement the GDPR regulations, or basically how they read the GDPR texts. So I think we are entering a phase where it will be easier to access data because, for example, Denmark had a very stringent interpretation of GDPR, which really prohibited a lot of data sharing. But now different legal entities at the universities have learned how to deal with GDPR in a way so that one can. What has happened is that this phrase that without considerable difficulty one can break a code. I mean, it's that opened up a lot in Europe. So basically, I think Leslie and others on this call, it will be easier. I think people are getting there now. The pragmatics of GDPR is starting to appear. So I'm hopeful that this will resolve. But it has been a struggle, and especially these types of cross-Atlantic collaborations have been harder to do for a number of years. And of course, that's a big problem that we need to fix. So it's an important feedback so that we don't destroy the opportunities of helping patients with doing research by regulations that are not helpful at all. And I agree with what you said. They have not increased the protection. They have just for a while made it harder. But I think this is going in the right direction now. And I know, I forgot to say, EU has formed a committee to look into this also, so to harmonize how different member states are implementing GDPR, so that it will still be possible to do international research.
Do you or other panelists want to put anything else on the table as being potentially the rate limiting step in this kind of work?
I could just make a general comment since I didn't bring it up in my presentation, but in the field of neurodegeneration, validating surrogate biomarkers, it has been so hard because of the conundrum we are in at the moment, or catch-22 situation, that we have no treatments that give us a clear clinical benefit. So we have lots of diagnostically validated biomarkers. It has actually been quite productive, but we are still lacking this piece to relate these biomarkers and changes in these biomarkers to a clinically meaningful endpoint. And the whole field is waiting for this to happen. And I think when we will have data where there is a clinically meaningful endpoint in a study, I hope this will be the key that unlocks the situation we are in at the moment. And then I hope that we will soon be able to use the biomarkers we have at hand. Some of them I showed data on, and then perhaps improve the design of clinical trials and the interpretation of clinical trials further. But at the moment, we are really in this situation that all biomarker researchers and pharma companies and regulators are looking for this link, of course, to a clinically meaningful endpoint.
I can add one more point, is that I think having a longer term follow-up on participants would help us to understand what really happened and then to have better estimates of what happened over a shorter time. Studies are often ended early for perceived lack of benefit or other issues, but having information on them afterwards would really help. I understand there's a lot of complexities if you're following people after the endpoint of a trial, but erring the side of longer information provide a lot more information for us. We talked about, even without the context of a trial, but within registries that are set up at the beginning to at least ascertain endpoints could help us as well.
Yeah, we're not so good in drugs at setting up registries and getting long-term data. And of course, companies are not going to be happy, I think, doing this some other way. So that's tough. Other thoughts from folks on, what are some of the big hurdles here? Okay, not hearing another candidate. I think we've probably identified what the largest hurdles are. Are there the second question we were posed was, to identify characteristics of programs that had a good track record for evaluating surrogates. Do we think there are such groups? I mean, Leslie, you've got one under your belt. Maybe a second one under somewhat further behind. But are there groups that actually have a track record here?
I could just mention that in the field of neurodegeneration, what has been so clear is that the big cohorts that have been built in the US and Europe, and now we also see them in Asia and South America, with cohort studies with repeat samplings, longitudinal follow-ups, relatively long studies, since the neurodegenerative diseases often have a long continuum. Those have been so extremely important to validate the biomarkers we have at hand for neurodegeneration, amyloid pathology, and tau pathology. And I think that has really set the stage for eventually being able to find biomarkers that will work as surrogate biomarkers in clinical trials. But still, we lack the drugs. But I think the scene has been set for seeing such results, and I think it will be quite easy to interpret or to provide the link between the biomarkers and the clinical endpoint once we have a drug that gives this type of effect that is then regarded clinically meaningful. So I would really like to emphasize that all this cohort building that has been going on over the past decade, I think that has been essential, and also being able to compare biomarker candidates across modalities, imaging, fluids, and clinical neuropsychological test scores, and genetics also, actually.
Okay, Eliza?
Yeah, and you were asking about good groups. Just to comment first, the history actually of our work with Leslie's group, I'm going to argue, and Leslie may push back, that, some of our early discussions, when I talk about early discussions, I'm probably going back maybe almost 15 years. I think, Leslie, that's about when I joined the agency, and maybe when, at least I got involved in some of these discussions, is it took a while for, frankly, I think maybe for, your group to understand what we really needed. And now I would say that, you've advanced the science and really forced us to think, about ways that people, that we validate surrogate endpoints, et cetera. And so in terms of, I really think there's this big education piece. And in terms of other examples that have been successful that I've been involved in, I've had successful examples out of the Kidney Health Initiative, where we sort of, and even before that, had met with the patient advocacy group, and early on, had spoken to patients and their families for rare disease about sort of the data that could support a conclusion about, a surrogate endpoint and biomarkers and how we thought about them. And then had this other group where, convener, that could bring us all together, the academics, the patients, the regulators, to really have, these meaningful discussions. So, a private partnership like that. But I also think, the Critical Path Institute is really set up well, to be this type of convener. And I think, we've heard about some recent initiatives, particularly on the rare disease side, where I think we're really going to be able to bring together the data and, of course, the necessary expertise. Because I think, again, a lot of it is sort of a translator is what we need, right? We sort of understand or have, fortunately developed, further thinking about the data that are needed to validate surrogate endpoints. But we need someone who understands us, but also works well with pharmaceutical companies and the larger community. I think if we could do a better job of explaining all this, not just to industry or academics, but even patients, that would be great.
I probably shouldn't say this because I have a highly biased opinion. I think we're doing a pretty good job, but I don't think it's really the organization that is going to determine the success of any effort. I think it's probably who is coming to the table to help whatever organization it is. I think it would be short-sighted to just have one stakeholder going through an endeavor of trying to validate some sort of biomarker, as opposed to making sure that all stakeholders are involved in whatever the design is as early on as possible. And I think that's very consistent with what Elisa was saying. But one approach that we've tried to take in our particular validation efforts around ctDNA is that just that approach, have all of the stakeholders involved. We have not asked simply the companies that have donated their data into this initiative to hand over the data and walk away. They've been a part of designing the statistical analysis plan, which I think is beneficial. They know their data the best. We do have an independent analysis center that does all of the, actually does the meta-analysis for confidentiality purposes. And that was important to make sure in order to get the data in. But it's been a strong collaboration with, experts from the FDA, from the clinical research community, the NCI, other organizations to help design and make sure that all of the different questions that each of those stakeholders might have are hopefully being answered. And we're designing up front how to try and answer those questions the best we can based on the data that's available for this initial prototyping concept. And I hope that in the long run, we know that this sort of retrospective type of approach is going to help establish the methodology. It's not going to be the definitive around the utilization of circulating tumor DNA as an intermediate endpoint. But I hope by doing this with many stakeholders at the table, it will provide the methodology that then can be put into a prospective trial for subsequent validation more readily, because the people who would then be putting that into their individual trial will be familiar with the methods and have the confidence in it because of the stakeholders that came together to design it in the first place.
Yeah, that's good. Henrik?
Yes, is it okay if I ask a question to Nicole? I was thinking when listening to your presentation, Nicole, that you have so many drugs that are super effective, and then you have also the biomarkers, and you have been able to link biomarkers to drug effects in such a nice way. How important was it with the first studies with effective drugs to be able to make this type of link between a biomarker change and a clinical endpoint, and did it unlock your field sort of? Is it a reasonable hope that this will happen in the field of neurodegeneration from your point of view?
Yeah, Absolutely. I guess I would say, hopefully learn from what we did well and what we didn't do so well. I think we, one of the things that I think and I think you're actually in your field a much better position is that, a lot of our early trials didn't necessarily integrate, some of these biomarkers. And I think even, once we had a little bit more understanding of how to use them, it took a while for us to standardize them across the board as well, how we are assessing them, such that, then when you're later going on to, assess the adequacy of the biomarker to serve as an endpoint in these clinical trials, if it's used entirely different ways in the subsequent trials, it's hard to really, collate that data and accurately assess it. I think, standardization as early as possible is something I think we could have done potentially a little bit better, in our area. But I think and I would just echo everything that Jeff mentioned. I think, perhaps learning from some of those mistakes, the broader community has really come together to try to, have a standardized approach moving forward. And a lot of discussions about how do we, move this benchmark in each of the various diseases and other, assays in the oncologic space. But I think, it really just sort of underscores the need for having broad, stakeholder involvement, from industry, academia, regulatory agencies to sort of, come together on what needs to be collected, when it should be collected, and standardize some of those features, such that then at the end, wherever you end up, you have data that can be reasonably collated.
Thoughts from people about specific things we can do better, either in terms of building infrastructure or adjusting the way we interact with regulators. What should be evolving towards here?
I really like this idea of communication. There's a lot of two-way communications that happen, say between the sponsor and the regulatory agency, and that's happened multiple times. How can everybody learn from all of those? I understand they're private and they're confidential, so I don't have an answer, but I think there could be a lot of learning. And I think part of what we've done in our group is try to develop ways in which that as part of this ongoing funding, we have these webinars twice a year. And I'm sure you struggle with this too, that the Duke group who organizes this, do you see everybody on the videos? And then it's hard for participants to collaborate. So last time we had a Zoom with everybody's faces. I said, please ask us a question. We want to talk to each other. Nobody actually asked questions, but I think that we're trying to develop a forum for that. And virtual has good things and benefits. Everybody can, and harm, or limitations. Everybody can be at home, and so they're more likely to participate. But it's hard to have those casual conversations that I think in meetings sometimes you can have, and that could allow for more of a community feel across people with similar interests.
Yeah, I remember in-person meetings. with fondly remember.
They're good.
Maybe someday.
Yeah.
Other thoughts from people?
I think it's really important to try and figure out where where the common ground can be and where sort of the mutual priorities are. A lot of times across different clinical development programs, the study design is going to be tailored to that specific molecule and, respectable considerations that need to be given to design for the success of that trial. So trying to figure out sort of what common elements can be pulled from different trials in advance with some of these data aggregation efforts is really important to try and improve the standardization of the approach. That's not to say there shouldn't be the flexibility designed within the individual trial to address specific concerns, but trying to figure out, I think, at the outset where data can be leveraged will help for some of these aggregation efforts, because unfortunately, when many organizations are designing their individual trials, it's not with the goal of sort of ultimately aggregating and validating some sort of a biomarker in these instances. Oftentimes you're sort of left with the data that you have by bringing the willing data partners to contribute the data together. So really trying to align on that up front, I think, could be helpful into the future to figure out where there are standardizations that can be put into place and where they may not be able to happen.
Yeah, I agree. I think there's a real opportunity for disease-specific interest groups, patient or investigator-driven groups, to try to drive that. To have some forethought, have some plans to try and make sure that as development proceeds in a particular area, that we're doing the best we can with collecting the extra information we want and trying to make it available academically to facilitate this. Other closing thoughts? We have 2 minutes left here.
I guess I would just add, and I think we've sort of all mentioned this, is just bringing as well, a little plug, bringing the regulators into the discussion early. Oftentimes when you're thinking about development of surrogate, oftentimes there's this thought, well, we don't have everything together yet. We don't really know how this is going to look or whatever at the end. And I think even in those formative stages, when thinking about assays and how you're going to look at them, or how you're putting together the meta-analysis, if that's what you're doing, et cetera, what trials, the FDA or the specific review divisions can really provide input and guidance on, what would be most helpful, from a regulatory, clinical trial perspective as well. So I just would encourage, including regulators in those conversations early on as well.
Since people have to drag us along anyway,
yeah,
they should get started early on that part. Steve, any other comments about what you think we can do better?
Well, I was thinking in my mind when you said, Norman, about I think advocacy groups have an important role. They're a point of continuity. I mean, they're in it for the long haul. They don't drop in and out of a certain area, and they have a real vested interest. And so I think best practices and what Jeff was speaking about, I think his organization is certainly way up there. A lot of best practices to share and get people stimulated early on to see the value in supporting these efforts. I think that's very important.
Okay, very good. Thanks everybody. Been an excellent set of presentations and interesting discussion. We're going to move into a break at this point and then restart 15 minutes from now.
Okay, welcome back everyone. We're going to resume our discussions with session 4, which is Beyond Surrogate Endpoints, Other Ways Translational Science Can Support Drug Development. And in this session, speakers and panelists will present use cases beyond surrogate endpoints. And the discussions will highlight how translational research guided the design of shorter, smaller, and more efficient clinical trials and help minimize risks to meeting efficacy and safety standards. We'll have 3 great presentations, which will all touch on different applications of translational approaches beyond the use of surrogate endpoints. Our first speaker will be Dr. Leslie Gordon, who is the Professor of Pediatrics Research at Brown University and co-founder of the Progeria Research Foundation. She will be presenting on a Hutchinson-Gilford progeria syndrome case study. Thank you.
All right, thank you everybody. It's been a wonderful, wonderful meeting and I hope I can add a little bit to it. I've been asked to talk to you not about biomarkers necessarily, but about some of the lessons we've learned in the field of Hutchinson-Gilford progeria syndrome to bring us from preclinical studies, cellular and mouse, into the clinic in this ultra-rare disease. Next slide, please. These are my disclosures. Next slide. And these are the children that we are trying to save, children from all over the world with progeria. Next slide. Progeria is a premature aging syndrome. It is an ultra rare disease. It is autosomal dominant genetic disease, which I'll talk to you a little pre and post about how important that's been for us. And the children all die of premature atherosclerosis, the same kind of atherosclerosis that you see in 60, 70, and 80-year-olds. And without treatment, they pass away at an average age of 14.5 years. Next, please. Now, these are some of the clinical signs of progeria. It is a multi-system disease. I'm going to concentrate in my case study on the cardiovascular aspects, but what you see here on the left is that they have severe failure to thrive almost from almost birth, starting at a few months of age, and that it affects multiple systems. But the mouse model studies and the human studies that I'm going to concentrate on focus mostly on the cardiovascular aspects of this, since that's what causes the children to die. Next slide, please. So the cardiovascular disease includes a progressive global atherosclerosis leading to heart failure and strokes. And what you're seeing here is a 5-year-old with a carotid artery complete obstruction on the right. Next. But what I want to show you also is the pathology of disease, because I'm gonna tie this in with the mouse models that I'm going to show you. The children develop calcific plaques, which is not easy to generate in any mouse model, but they have a thickened fibrotic adventitia, and the media of their blood vessels becomes, as the disease develops, almost devoid of cells, of smooth muscle cells, and that's replaced by extracellular matrix deposition. So that's what you're seeing up here on the top, and then non-HGPS on the bottom is an older individual with atherosclerosis. Next, please. Okay, and this is two of the ways that we measure this in the clinic. And this is going to, especially the pulse wave velocity on the left, be very informative for the presentation about the mouse models. Carotid femoral pulse wave velocity is essentially the rate at which blood can get from the neck to the femoral area. And when you have a stiff pipe, when the vasculature is very stiff, as in progeria, the blood has to move a lot faster, and that increases the pulse wave velocity. So a higher number means a stiffer vessel. And you see the normal range here, age matched, is 5 to 6.5, and all of the circles of children with progeria untreated, and their levels are very, very high. And this is very important measure in the clinic, because this is one of the major outcome parameters that we use. And you're seeing this sort of pathologically by echo on the right as brightness. Vascular wall brightness on the right here versus the left means that there's a lot of dense extracellular matrix in the vessel wall. Next, please. Well, we knew something about progeria before the gene mutation, but the gene mutation discovery opened incredible doors for us in regard to being able to design, get support for preclinically, and then move into the clinic. So in 2003, Francis Collins's lab, in collaboration with us, led by him, discovered the gene mutation for progeria. Next slide, please. And this is essentially what it is. The gene mutation lies in the gene LMNA, which produces a protein called lamin A. And lamin A lines the inner membrane of nuclei. It binds to chromatin, affects transcription. It has both structural and cell signaling effects. And importantly, it's expressed in all differentiated cell types. But progerin, which is the causal protein in progeria, not the lack of lamin A, but the presence of progerin, is a shortened protein, abnormal lamin A protein, that also has problems with its post-translational processing. So we've got an abnormal protein that is short and not processed properly, present in the nuclei of differentiated cell types. Next slide, please. So after the gene mutation was discovered, we could diagnose children properly. We didn't have to deal with misdiagnoses like we had in the past. And we could produce progerin-producing cell models and mouse models, and in an effort to move us into clinic more accurately. Next, please. Here I'm showing you sort of the hallmark, and there are many ways to measure, to find abnormalities in cells, but this is our sort of hallmark sign of what happens to cells, culture cells in progeria. There is nuclear blebbing. As you can imagine, the abnormal protein in the nucleus is tethering and pulling in ways that cause blebbing. It increases with passage number, and it causes the cells to die early. Next, please. But this is a slide showing you the sort of hallmark mouse models. Now, there are a number of mouse models for progeria, but I'm just going to talk to you about the two that I think have really guided the field in a powerful way. They are both progerin producing, and that I think is very important. One was produced in the Collins lab. It's a BAC transgenic, bacterial artificial chromosome transgenic. So the BAC has a human gene in it, producing the G608G mutation that causes progeria. Progerin is produced in these mice. And then secondly, there's a knock-in model where the mouse gene is mutated. That's a G609G, so it has sort of a mouse progeria. Progerin is also produced in that. And they're different from each other, but they complement each other in wonderful ways. And the G609G was produced in the Lopez Otin lab. They're both small. They both develop cardiovascular disease, but at least early on, and even medium on, they don't have plaques. They both die early, but as in a lot of mouse models, we don't know exactly why they die. And as I said, they both produce progerin. And what you can see here on the right is the cardiovascular disease. The wild type aorta on the top looks very different from that on the bottom, which is basically devoid of cells. It's late on, and a lot of the smooth muscle cells have died off. Next slide, please. Okay, here I'm just showing you the post-translational processing of lamin A on the left and progerin on the right, just to demonstrate that our first clinical trial treatment ever for children with progeria was born out of the gene mutation understanding and the post-translational processing of the protein. So it's biologically based. It's called lonafarnib, and the category for this drug is farnesyltransferase inhibitors, or FTIs. And that's what I'm going to focus on in our first sort of studies that bridge to trial. Next, please. Okay, so this is sort of a summary of some of the data that brought us to trial. On the top left, you're looking at fibroblast cultures treated with a farnesyltransferase inhibitor, and that normalizes the nuclear shape. And on the bottom, you're looking at the vessels, the blood vessels from a mouse model that is progerin producing, treated with an FTI, not the one we used in clinic. And on the left panel, you see an untreated aorta. In the middle, treated mouse aorta, and on the right, a wild type. And obviously, the treated mouse is in much better shape. Next slide, please. All right, so what you're seeing here is the clinical trial results. On the left, we've got pulse wave velocity drastically improved by the end of the study. In the middle, echo density improved, and on the right is a Kaplan-Meier curve with the blue untreated and red treated children, and obviously, and there was an improvement in survival in these kids. So that's the story of great translation. Next slide, please. Next. Okay, but now next. I guess we're going to keep going down. Why don't you keep going till all of the treatments are showing? Terrific. Now, what I want to show you here is just that we've run several types of clinical trials, all sort of based on the pathology of how the abnormal protein is produced. After the lonafarnib study, we added 2 prenylation inhibitors, a little more upstream, but biologically based. And we added pravastatin and zoledronate. Next, go to the next, keep going to the next slide. Keep going, there we go. Now, just in a nutshell, what I'm going to show you here is a bit of weakness, a lesson learned along the way. We had some cellular studies, as you're shown up here on the left, showing that yes, nuclear shape was improved with the bisphosphonate and statin. And on the right, a mouse model, that's a progeroid mouse model, but not a progerin producing mouse model that was used at the time, because that's kind of all we had. And so this ZIMST24 mouse model is a bit different from progeria. It doesn't develop heart disease, cardiovascular disease, and it does fracture, get spontaneous fractures and neurological deficits, unlike those with progeria. And I think that's pretty important, because that may be the reason why one of the main reasons why they die, maybe lots of fractures, and bisphosphonates would have helped that. And in the human trials, we did not see a benefit of these 2 drugs over and above lonafarnib. So for us, that was a bit of a lesson learned in which model should be used or could be used, although this is a great animal model, maybe not optimal for progeria. Next slide, please. Okay, so I'm going to flip back to one of the progerin producing mouse models and talk about how optimizing and communicating and optimizing care for that model has really changed us and helped us a lot. Only recently, in the last couple of years, Jay Humphrey from Yale started using a soft-based chow, introducing a caretaker mouse, and was able to increase the average lifespan of these mice significantly. And this allowed the cardiovascular phenotype to actually worsen and become much more similar to that observed clinically in patients. And the vessels got very stiff, but also we saw vascular calcification. Next, please. And so what you're seeing here on the left is the ability to measure carotid femoral, measure pulse wave velocity, a measure of pulse wave velocity in the original mouse model, not at 103 days, the average at first, but after 168 days. Look how high that is. So the animal husbandry and the care really helped. And then what you're seeing on the right is that it did improve with lonafarnib treatment. So this is a perfect now way to say, oh my gosh, this improvement is going to help us get to the clinic with drugs that have more potential for benefit. Next, please. So, I want to finish out by concluding and providing a couple of suggestions for maybe centralizing resources and using these 2 examples that I've shown you in an effort to optimize preclinical studies across an entire field. One is very inexpensive and one is very expensive. So here's the first one. Essentially getting the word out, sharing information that might be really important, the devil's in the details, collecting and distributing best practices and guidance for basic scientists through scanning new publications, investigator surveys, and then doing email blitzes or having a resource center like we do at PRF in our cell and tissue bank. Next, please. And then finally, this is a big project, but centralizing disease-specific animal testing could be key to an entire field. We haven't done this yet, but think about if the primary animal models are all available in the same place, treated in the same way. You can choose the candidate interventions. The investigators can be very involved. Survival studies would be very consistent, pathology, and some basic phenotypes. And then tissues or other things can be sent to investigators, even the mice, for specialized analysis. And I think that's, it is an expensive way to go, but I think that it could be very powerful in goals to make things consistent and get to trials. Next, please. In the field of progeria, we have a long way to go. We are still learning these lessons. I've just shown you a few, but we have small molecules and RNA therapeutics and genetic editing that we're working on. And we're using these lessons to try to get us into clinical trials to help and save these children in a very, you can only, with a very rare disease, you can only do one, maybe two. We've never done that, trials at a time. You have to be really careful, obviously, in what you choose. Next, please. Thank you very, very much. And again, I've really enjoyed this meeting. It's been fantastic, and I look forward to the discussions.
Okay, thank you, Leslie. I'd like to now turn to Dr. Estelle Merer-Berger, a senior translational safety leader at Roche. She'll be presenting on optimizing early clinical investigations by increasing the predictive value of non-clinical activities.
Thank you, David. It's a real pleasure for me to be here today, and I'm very thankful about the opportunity to share with you this translational package, which supported the entry into human of T-cell bispecific, currently tested in a phase 1 dose escalation in AML patients. The particularity of this package is that it was built with human and patient model systems as opposed to animal studies. And I will try to highlight through the presentation all the efforts made and particularly our lessons learned. Next slide. So let me tell you a little bit about the molecule. So here we are going to be talking about a T-cell bispecific. These molecules are matchmakers between a cytotoxic T-cells and a tumor cell. And the matchmaking will lead to the lysis of the tumor cell. We at Roche have a molecule antibodies that have a 2+1 format, which means that we target twice the tumor, and we engage the cytotoxic T cell via the CD3 epsilon binding. And here, generally, these CD3 bispecific target oncoproteins, but here the particularity of the case I'm going to present today is that we target not a protein, but a peptide that is presented by an MHC class 1 molecule. So we target complexes of oncopeptide and MHC class 1. And here we target a peptide that is coming from a oncoprotein called Wilms tumor 1 that is going to be presented by HLA-A2. WT1 is an intracellular transcription factor that is overexpressed in leukemia, in AML and ALL, and solid cancer. In adults, this oncoprotein has an expression that is restricted to a few tissues like kidney, testes and ovaries, a few mesothelial cells, and bone marrow. Next slide. So here, these molecules have the particularity to target MHC peptide complexes, and this bears certain challenges, especially for safety. When you develop this molecule, the non-clinical safety assessment has to be done a little bit differently. Why? Because these targets are exquisitely human. And this invalidates the conventional animal testing approach because these molecules lack cross-reactivity in animal species. Also, the entire toolbox actually of the toxicologist is not applicable to these programs. And in addition, as an additional challenge, you have an increased risk for off-targeting cross-reactivities, because by targeting only a 9-mer peptide, you can imagine that there are lots of promiscuous peptides that will also be presented by HLA-A2 that could eventually be bound by the molecule if this is not exclusively selective. So this is about the challenges faced, but there were also lots of opportunities. There are opportunities for us to think about different ways to build on a PK/PD safety translational package with human and patient-derived material, and actually test whether or not we can increase the predictive value by predicting from human to human. Next slide. So this is our non-clinical strategy for entry into human, and it was entirely based on NAMs. And from this approach, we derived the therapeutic index, the starting dose, and the pharmacologically active dose range. So in regards to safety, to ensure that the molecule is targeting selectively the complexes we want to, we build a strategy around 3 main pillars that we used iteratively to potentiate the risk identification. Because as said, with this molecule, every time you bind a healthy cell, you will kill it. So the first pillar was looking at testing the molecule in vitro in a broad range of human 2D, 3D models. The second pillar was a ligandome elucidation with human vital organ lysates. And in this pillar, we used the clinical candidate to actually fish potential off-target from a vital organ lysates. And we complemented the package with a study in HLA-A2 transgenic mice. The safety risk identified were integrated. So the qualitative aspect was integrated with a quantitative assessment, a PK/PD assessment to define a therapeutic index. And by saying that, I already give it away that we had a flag in our in vitro testing package, and the entire PK/PD safety data that we produced were used to define a starting dose and a risk management plan. Next slide. So this is our first pillar, basically. We did the risk identification using human 2D, 3D in vitro systems. As you can see, we had a broad diversity of different human primary cells that we tested. And the way we test these T-cell bispecific is by exposing a co-culture of human primary cells with allogenic or autologous PBMCs. So we test a broad range of our TCB, and we also look at a time course. So we evaluate the effect across a broad time course. We monitor different endpoints, and these endpoints are also looked at in the context of this time course and are integrated in a PK/PD model, all together to basically look at the effect. And as you can see here, I already flagged in red the liver spheroids. So I will be talking in the next slide about the flag that we had when we tested our molecule on liver spheroids. Next slides, please. So here, and you can click another time. Thank you. So here, when we tested our WT1 TCB on liver spheroids, and we knew that the target was absent from these spheroids, we could see a minimal lysis in these spheroids. As you can see here, we monitored LDH as a surrogate for lysis of cell, killing of cell, AST as a biomarker for hepatotoxicity, and IL-6 as cytokines that are released after the TCB elicit its effect. What was very important for us is to control this system with different negative and positive controls. So the negative control, we use spheroid only, we use the vehicle. The negative control that was used was the CD3 the CD3 arm of the bispecific without the tumor targeting arm. Here, I apologize, in blue is our positive control. It is called ESK1. It's a molecule that is targeting the WT1 peptide presented by HLA-A2 as our WT1 TCB. But this molecule is known to be unselective. So it's basically killing every primary cell co-culture with PBSC that we can think of. So next slide. When we realized that we had a flag, the question was, and now what? What are we going to do? So when you do an assessment based on in vitro data, you face a certain level of complexity in translating these in vitro effects into into the context of a human body. It's a little bit challenging to assess quantitatively, actually, what it is all about. And when you have an effect, you need to define the threshold of significance for your effect. You also need a therapeutic index, and you need eventually to look at causality and exacerbating context. Next slide. So we did that in a lot of different follow-up assays that we run, and to really look at consistency of effect and define our threshold for significance, we looked at different conditions, HLA-A2 negative versus HLA-A2 positive. We took allogenic PBMC, but we also run the same experiment using autologous PBMCs to check what level of alloreactivity was intrinsic to the system with allogenic. We looked at different biomarkers, LDH for lysis, alpha GST as a very strong marker for hepatotoxicity, an early marker. We looked at interferon gamma, granzyme B, and by evaluating the full package, we concluded that based on our dynamic in vitro killing assay and assessment, we had a mild but consistent signal at 1 microgram per ml. So next slide. So as I mentioned before, the grading of severity of an effect in the context of an in vitro model is not an easy thing to do. So we decided to go after potentially the causality of this signal, and we went after the identification of the off-target. And to do that, we run, as I mentioned before, the interactome assay, where we used our WT1 TCB clinical candidate to fish the off-target from human primary hepatocyte lysates. From this effort, we isolated about 70 different peptides, knowing that this assay would lead to false positive. But we follow up with a lot of orthogonal testing, which led us to one epitope as the only potential causality of the signal that we were observing in the hepatocytes, which was an epitope coming from a cytochrome P450, the CYP8B1. And actually, to substantiate our claim that the effect was minimal at 1 microgram per ml, and to also substantiate the naming of the threshold as being conservative, we ran a synapse stability assay to look how stable the synapse formed with the targeted peptide, which is the RMF peptide from WT1, is compared to the off-target, the CYP8B1, because the killing potency or the killing efficiency is increased in function of synapse stability. And here we could definitely show that with the off-target, we had a less efficient killing process than with the on-target. Next slide. So this led us to further investigate the therapeutic, to derive a therapeutic index from the experiment that we run. So from a safety perspective, as I mentioned, we use the primary liver spheroids, and we use these ex vivo in vitro framework, and we decided to apply an harmonized experimental design to look at efficacy. And to look at efficacy, we use primary AML samples. And by applying this harmonized experimental design, we could evidence that we had a therapeutic window, and with a translational PK/PD modeling strategy, we could define that our efficacious range would be actually where we would have only very minimal effect on the hepatocyte. Next slide. So we leverage actually also the efficacy model that was used with primary AML samples for the patient-centric starting dose prediction. And actually, you know, classically, we use an in vitro MABEL approach to derive the starting dose. And this classical in vitro MABEL approach uses the most sensitive tumor cell line and the most sensitive readout to derive the starting dose. Here, we decided to go for a system that we found most relevant to predict. And ultimately, we managed to start as at a much higher dose of 150 microgram compared to a 5 microgram dose derived from a standard MABEL approach, which enabled to reduce the number of patients treated with subtherapeutic doses. Next slide. So really quickly, and we can move to the next one, our learnings. It's very important to understand your system and your testing framework. You have to leverage the strength of the system, but be aware of its limitation. In the context of our assay, when testing TCB bispecific, we use a very dynamic system. So it's very important to evaluate the concentration effect relationship throughout a time course as long as the system allows, and integrate all the data into what we call an area under the effect curve. So that's a very critical step. Next slide. It's also very important to understand what biological effect you are going to follow, and to follow it from the beginning to the end with different relevant biomarkers. And it's important to look at consistency of your signal. And next slide. Also, it's very important to try to use material for your testing that is relevant for the patient you are going to test the drug into later on. And here again, the patient-derived material enabled the balancing act to achieve a higher starting dose for this CD3 bispecific. Next slide. So ultimately, we received a broad regulatory approval for this package. We dosed the first patient in November 2020, and the entry into human dose predicted from these patient-derived AML blast and autologous PBMC was safe. So far, we have no evidence for liver toxicity up to a dose of 12 milligram, which is way beyond our 1 microgram per ml threshold. But also what we can say is that the patient-centric framework is of high predictive value for the estimation of the pharmacologically active dose range. Next slide, maybe just quickly an outlook, and then we can move to the next. So, and you can click through, please. So here we know by now that patient-derived material is of high value in the preclinical and translational development. But our next step in our vision is to leverage this ex vivo testing in the phase 1 clinical trial to identify responders and predict efficacious dose. And next slide, this is what we are currently doing in the context of this phase 1 dose escalation. We are looking at individual ex vivo versus clinical in vivo PK/PD. And we use our primary AML model and the testing framework on the the bone marrow samples we get from the patient enrolled in the trial to predict the ex vivo potency. We combine that with the individual PK, and we actually in this pilot try to understand to which extent this ex vivo model plus testing framework could predict the right dose for each patient individually by comparing actually in vivo data in the context of the clinical trial. Okay, I'm going to stop here and thank you very much for your attention, and I'm welcoming any of your questions.
Okay, thank you very much for the wonderful presentation. Our final presentation will be from Dr. Christine Garnett from FDA, clinical reviewer and team leader in the Division of Cardiology and Nephrology at FDA, the lead for the Cardiac Safety Interdisciplinary Review Team. She will present on pediatric extrapolation for pulmonary arterial hypertension using a physiologic biomarker of pulmonary artery pressures. Christine?
Well, thank you. So my presentation will be a little bit different from what you heard from the previous two in our session. And I will focus on clinical translational science, whereby a bridging biomarker can be used to extrapolate efficacy of a drug that's already been approved in adult population to a pediatric population with the same disease. Now, the approval of new drugs for use in children follow the same high standards as that of adults, requiring demonstration of substantial evidence of effectiveness as well as a favorable benefit-risk profile. However, there are ethical, physiological, and statistical challenges in conducting clinical trials in children, and approaches that leverage as much clinical data from this reference population to the pediatric population are important to reduce extensive testing in children. And this is why I will focus in my presentation on this pediatric extrapolation approach, as well as the bridging biomarker. And I will present a case study using PAH. Just to have some, next slide, please. So just as a standard disclaimer that my presentation is not considered official FDA guidance. And I'd also like to acknowledge contributions and insights to this presentation by Drs. Norman Stockbridge and Lynn Yao at the FDA, as well as Dr. Thomas Fleming at the University of Washington. Next slide, please. The pediatric extrapolation is an approach to provide evidence in support of effective and safe use of a drug in children when it can be assumed that the course of disease and the expected response to drug would be sufficiently similar between the pediatric and a reference population. Now, in my presentation, the reference population will be the adult population, but keep in mind that a reference population can also be another pediatric group. Next slide, please. So the factors that influence extrapolation will depend on evidence-based assumptions on the similarity between the adult and the pediatrics for the disease, as well as its response to treatment. So the first is evidence for disease similarity. This would include factors such as common pathophysiology, common disease definitions, as well as the course of disease. Evidence for response similarity would include factors such as similar pharmacology, similar response in clinical endpoints, as well as demonstrating a similar exposure-response relationship. And then there's the quality, the quantity, and the sources of existing data are also important considerations. So all these three factors will influence the choice of an extrapolation approach. Next slide, please. So the types of extrapolation approaches, you can think of them as reflecting a spectrum of dissimilarity and similarity of disease and response to treatment. So if there's a high confidence in the similarity of both the disease and the response to treatment, then pediatric extrapolation approach could include a pharmacokinetic study to show similar exposures in pediatrics to that of adults. Now, on the other end of the spectrum, if the disease is different, or if there's large gaps in knowledge, whether the response to treatment then the pediatric approach could include one or more adequate and well-controlled trials using age-appropriate clinical endpoints or surrogate markers. Now, somewhere in between the two ends of the extrapolation spectrum is the use of a bridging biomarker, where there is high confidence in the disease similarity, but there remains gaps in knowledge on whether there is similarity in response to treatment, and perhaps there are limited age-appropriate clinical endpoints. Next slide, please. So a bridging biomarker is a response biomarker that is supported by strong mechanistic evidence and is expected to be correlated with a clinical endpoint intended to assess clinical benefit in clinical trials. And the use of a bridging biomarker in pediatric extrapolation is distinct from other roles of other biomarkers. Its role is to extrapolate the efficacy from adults to children for drugs that have already demonstrate effectiveness in the adult population with the same disease. Now, a bridging biomarker is more predictive of efficacy or the disease than pharmacodynamic biomarkers that are usually used in pediatric development programs for dose selection, as well as maybe demonstrating similarity in the disease or response. But it's not. But there's less. It's not a surrogate endpoint. There's less confidence that it directly is a measure of how a patient feels, functions, or survives. So next slide, please. Now, to justify a bridging biomarker as a replacement endpoint in pediatric trials, the biomarker should meet 5 criteria, which I have put on this slide. The first criteria is that the pediatric and the adults have similar disease. The second criteria is that in adults, the drug has substantial effect on both the clinical endpoint and the biomarker. The third criteria is that the treatment effects on the bridging biomarker capture the effects on the principal causal pathway through which the disease process meaningfully influences the clinical endpoint. The fourth criteria is that in adults and also in the pediatric populations, that the treatment doesn't have any important unintended effects on how a patient feels, functions, or survives that are not captured by the bridging biomarker. And then finally, the fifth criteria is that by proper statistical analysis, that the treatment's net effect on the clinical endpoint is consistent with what would be predicted by the level of the treatment effect on the bridging biomarker. This is very similar to the Prentice criteria, but is not as stringent. So if these 5 criteria are met, then establishing a meaningful effects on the bridging biomarker in the pediatric setting would provide sufficient evidence to extrapolate the efficacy assessment from the adult population to the pediatrics, that the drug would have a meaningful effect on the key clinical endpoints. So next slide, please. So now I'll move into my case example. And this case example is how pediatric extrapolation using a bridging biomarker was used to approve the first drug for pediatric pulmonary arterial hypertension. So conducting clinical trials in pediatrics that have pulmonary arterial hypertension, I'll just call that PAH in the presentation, is very difficult. First of all, PAH is a rare disease and there's limited patients who can participate in clinical trials. It's also very difficult to conduct placebo-controlled trials because there is a prevalence of off-label use of drugs that are not approved for children, but are approved for the adults. And so it's very difficult to have patients enroll into a placebo-controlled trial. And also in pediatrics, there's a limited number of reliable clinical endpoints. A lot of the adult drugs for PAH were approved using exercise testing, such as the 6-minute walk distance. But this is really not a practical endpoint in pediatric PAH because you really can't do exercise testing in the very young children or children who have developmental disabilities. So next slide, please. So pulmonary arterial hypertension, again, is a rare disease with a diverse cardiac, pulmonary, and systemic disease that has significant morbidity and mortality. And pediatric PAH shares very similar disease with the adults, and clinical experts have agreed that the disease is similar enough for pediatric extrapolation. And so pulmonary vascular resistance is a hemodynamic measure of the pulmonary arterial pressure and cardiac output. And PVR is on the causal pathway through which the disease process impacts how a patient feels, functions, and survives. Next slide, please. So there are 10 drugs from 4 therapeutic classes approved in the US for treatment of PAH in adults. The 6-minute walk distance is a submaximal exercise test that has been used for most drug development programs to establish efficacy for new therapies in adults. And so what I'm showing you on this slide is that there is a correlation between the decrease in the PVR, which is on the x-axis, and an increase in the walk distance, which is on the y-axis. And this is a pooled analysis of 9 approved drugs from 12 placebo-controlled trials. And each individual point on this plot is an individual patient. The red is from the treatment groups and the blue is from the placebo groups. And so what I wanted to do next, next slide, is just to show you, this is a pooled analysis. This next slide now shows you 2 forest plots. The plot on the left compares the relationship between PVR and 6-minute walk distance across different drug classes. And what you can see then is that across all the drug classes, there's a highly overlap relationship between the different classes, as well as overall, which is in red. And if you compare, and if you look to the plot on the right, this is looking at the same relationship between PVR and 6-minute walk distance for individual drugs. And sometimes this is an individual study, and sometimes these are pooled studies across a couple trials. And what you could see is there are also a good consistent relationship, and even with bosentan. And I've highlighted that by the red box. So we'll go and look at more into bosentan in the next slide, please. So in adults, treatment with bosentan had a significant effect on both the 6-minute walk distance and improved it by a mean of 35 meters. And it also had a significant reduction in PVR. And so when we looked at a statistical model where we were trying to evaluate the percentage of the treatment explained on the 6-minute walk distance, we found that the model showed there was about 50% of the net effect on the 6-minute walk distance could be explained by changes in the PVR. And so this intermediate result was further reinforced just by the biological importance of PVR in the disease, where we know that it's influenced by both the pulmonary arterial pressure and cardiac output. Now, in adults, as well as in long-term studies in children, there was an absence of unexpected deaths or serious adverse events with bosentan. So we don't believe that there was any unintended effects of bosentan in PAH populations. So overall, the adult data showed that PVR can be used as a bridging biomarker, as it met the 5 core criteria. So next to show you what bosentan's effect on PVR in children with PAH. So we'll go to the next slide, please. So in a small uncontrolled study in pediatrics, with pediatric patients with PAH who were taking bosentan, efficacy measures were collected. Because it was a small study, there was only 19 patients, pediatric patients. They did collect some exercise capacity endpoints in there, but it really didn't show any improvements in exercise, but that's not unexpected because the study was so small. But there was improvements in the majority of the hemodynamic measures, including PVR. Now, bosentan significantly reduced PVR, and that's shown in these box plots on this slide. So the reduction in PVR in children is shown by the orange box. And the magnitude of change or decrease in PVR was similar to that observed in adults who were taking the labeled dose of bosentan, and that's shown by the next box, which is in light blue. And you could see that both the bosentan treatment and pediatrics as well as adults were a lot lower than the placebo effects in both pediatrics and adults. And these are coming from different study in pediatrics. So this is the yellow box is a cross-study comparison with the orange box with bosentan. But you can see that the placebo response in both the pediatrics and the adult were very similar. So we didn't expect that, the pediatrics would respond differently than the adults on placebo. So based on this study, this very small study, again, it was only 19 patients with pediatric patients with PAH. The FDA concluded that bosentan's effect on PVR in children corresponds to a clinically meaningful effect on 6-minute walk distance. Next slide, please. So in 2017, the FDA did approve bosentan for the treatment of PAH in children based on this extrapolation approach using PVR as the bridging biomarker. And what I've shown on this slide is the bosentan indication in children. And which is highlighted in yellow and in orange, you can see that the labeled indication reflects the use of PVR as a bridging biomarker. So unfortunately, moving forward and trying to think about using this extrapolation approach to approve other treatments for PAH in children, we did come to a obstacle in that PVR is collected by right heart catheterization. And this procedure, just the procedure of the right heart catheterization, has been shown to cause serious adverse events as well as death in children. And so because of that, we're no longer using right heart catheterizations in pediatric trials as part of endpoints. I mean, it's still being used as part of the clinical course, but in clinical in the clinical part of it, but in terms of clinical trials, we're not using right heart catheterizations anymore. So there's ongoing efforts to be able to measure PVR or other hemodynamics, such as cardiac output by imaging techniques. And there are also efforts to develop endpoints for children collected from actigraphy. And I understand yesterday there was a question about the use of actigraphy, and I just wanted to try to address that question here. Is that actigraphy would be another measurement of functional assessment, or it would be a direct measure? It would be considered a clinical endpoint. And one of the reasons I haven't, at least within DCN, we haven't seen it as a endpoint is that we still don't understand the clinical meaningfulness of any changes in measures coming out of actigraphy. And so I think there still needs to be some more work done to be able to find that, an endpoint using the actigraphy. But if we did, it would be a functional measure. So it would fall under a clinical endpoint and not a surrogate endpoint. So just, can we go to the next slide, please? So just for conclusions, I just want to point out that bridging biomarkers have an important use for pediatric extrapolation, and a bridging biomarker is very distinct from other biomarkers that are used in pediatric development programs. It's not a pharmacodynamic biomarker, nor is it a surrogate endpoint. So it's somewhere in between. And to be able to establish a biomarker as a bridging biomarker for decision making, the biomarker should satisfy the 5 core criteria that I presented in this presentation. And I've used an example of how bosentan was used for approving the first drug in pediatric PAH. We also have used the pediatric extrapolation with another biomarker for approving Entresto for pediatric heart failure. And we did that using the bridging biomarker NT-proBNP. So we are using this type of bridging biomarkers in pediatric extrapolation at the FDA to approve drugs for pediatrics. Next slide, please. So I guess these are my references, and I will be looking forward to our panel discussion. So I'll turn it back over to you, David.
Okay, thank you very much, Christine. So I'd now like to welcome our additional panelists. We have Dr. Anthony Dermowitz, who is Vice President of Clinical Development for the Cystic Fibrosis Foundation, Dr. Lin Yao, Director of the Division of Pediatric and Maternal Health in the Office of New Drugs at FDA. So the panelists, please turn on your videos, and we'll have the full slide with all of the panelists shown here. The first question is going to be to Tony. Can you talk about the benefits of these translational approaches in drug development beyond the use of surrogate endpoints, or and especially for emerging genetic therapies?
I'm sure, Dave. I thought, speaking for some of the panelists, I think it was kind of interesting, when we heard we were in session 4 and it was the others. It's like, what exactly would that mean? But I think we've outlined, or the organizers have come up with 3 really good classes of different kind of others that are really important with the examples. I mean, we saw the bridging, just with Christine, the bridging and translational biomarker endpoint for pediatrics. We looked at with Estelle a lot of in vitro assessments and how they can help drug development. And then we heard from Leslie about the animal models and progeria that were being used. And each have a great place, and they're good for certain things, some more for others. Like, for instance, I'm a pulmonary hypertension researcher from 20 or 30 years ago. So in pediatrics. So I mean, it's obvious when you know what the causes of pulmonary hypertension are in children that it's different. The pathophysiology is different than a lot of adults. So you need these kind of bridging biomarkers to help support extrapolation. Sometimes you don't, because the pathophysiology is the exact same, and fortunately for CF, you don't need that. But I could see, in genetic therapies, needing these kind of biomarkers to be able to translate adult data into younger individual data as well. So I think it can be very important in that aspect. With the in vitro stuff, I think I kind of view that as the workhorse of the others, if you will, in that you can do a lot of things with the in vitro stuff that's going to be really helpful in de-risking and streamlining your program, as Estelle was kind of outlined if you looked at the cool stuff that Roche and she were doing with that stuff. So for a gene therapy, you can look at target engagement to make sure there's adequate engagement for the drug even before you start thinking too much else. You can select a study population or subpopulation if you need to. For instance, I'll just use the CF thing because that's where I'm from these days. There's about 2,000 genetic variants in the CFTR mutations. So not every drug you develop is going to capture every variant there. So, having these kind of in vitro techniques, whether it's electrophysiology data or something else, to weed down the responsive population is huge in that. You can establish proof of concept. You can establish a dose. Estelle was talking about dose establishment too. And, frankly, clinical endpoints, although critically important, can sometimes be very blunt instruments when it comes to dose separation. So having those kind of assays to be able to show there's a dose separation is huge. And then it helps all frame the risk benefit, determination at the end. The final thing was the animal model stuff. And animal models are, for genetic diseases and for other diseases as well, are like incredibly important for some. And the approved genetic type therapies that we have all that I know of have had fairly convincing, animal model data, whether it's, with what Leslie was saying, or whether it's with the SMA program that was done, or others. So they can probably do everything that the in vitro stuff can do, as far as that list that I was going through. Plus, we heard yesterday that it's the possibility for animal data to be a part of the confirmatory data package for a small small phase 3 program, if you will, with only one study. The problem sometimes with the high throughput stuff, and we see this as well, is that it's tends not to be a high throughput kind of system. So it's better to kind of move with what, at least first, what you can do. And so all three of these are really cool and key things to do. But the challenge is how do we promote or foster development of these things across different disease indications and sponsors, et cetera. And when I thought about that, and other people have thought about it too, because I heard it, during today's discussion a little bit in the other sessions, is, two words come to my mind, and that is sharing and transparency. And I'll tell you, the CF Foundation funds some drug development programs across companies or sponsors that might have, programs that you could view as kind of competitive, if you will, because they're in the same therapeutic space, whether it's an anti-infective or whether it's a genetic therapy or something. And, sharing is really hard. And, getting those people in the room, or not even in the room, just to have a conversation on common issues that are not really far down the road is incredibly hard. If it's a big pharma company, they tend to have a lot of resources. I'm not talking about you in particular, Estelle, but they tend to run things internally and have what I would call a consortium of one. If you got a small biotech company who doesn't have an approved drug and they're burning money like crazy trying to get their way forward, they're so interested in kind of guarding and protecting their intellectual property and being really quiet about things until they can get a hit on something, that they don't talk too much either. I think the cooperation comes in sometimes, I think, in this context with the ultra rare population, ultra rare disease kind of therapy programs where people need each other too much. And we saw that maybe a little bit with progeria. So that kind of makes me think about the transparency thing, and how we can, maybe greater transparency in certain areas can kind of move the ball forward a little bit. And one of them, yesterday it was brought up, is
hey, Tony.
Yeah.
I just want to make sure we maybe get to some other panelists also. We only have about 20 we have 20 more minutes of the discussion, but
okay. Well, I'll just cut it very short here and say, greater transparency. And the two topics that I thought about in the greater transparency aspect were, access to failed clinical trial data, even if it's placebo, to be able to share. And the other thing I'm going to call you guys out at the FDA is that kind of what I would call more intentional regulatory transparency. And I'll stop there and leave you hanging, because I think we kind of need to go on.
Maybe we'll see if we can circle back to that at the end, figure out what you mean by that.
A secret now.
Yeah, okay. So Lynn, I'd like to go to you next and ask if you can talk about how translational science approaches other than surrogate endpoints can support regulatory submissions for accelerated approval or traditional approval.
Yeah, thanks, David. And I think Christine gave a very, very nice example of how we're evolving and leveraging what we know. My key point here, regardless of whether it's an accelerated approval, whether it's going to be used as a bridging biomarker in a pediatric extrapolation approach, whether it's going to be used to support even just a population that could be studied or a dose that could be moved into phase 3. The key to all of this is really providing the data package that justifies this biomarker for that use. And so much of it requires the upfront work. And that's where I really feel like I'm the regulator here, okay? So to Tony's point, I'm not the one that's got to dig up the capital to figure out how to do this early work. But the reality is if you don't do the work early, you're not going to get it on the back end, right? You're not going to be able to validate a biomarker after the trial is over. You have to do that for your particular program. You've got to do that work up front. And even more importantly for children, if you want to use an extrapolation approach, that requires that you think about those biomarkers or those clinical endpoints up front and get that exposure or dose response in the adult program that will support matching it in a pediatric program. So my plea to everybody is to think early. And I don't know how you work together because we've tried this many times. I appreciate CPAC. I appreciate groups like the Cystic Fibrosis Foundation. I appreciate, international collaboratives. But a lot of this work, if it's not done by a consortium, I don't think it's going to fall on the drug companies because they're going to do it for their program. I'm not sure that they're so interested in doing it for their competitor. So, I would love to hear more in the future or maybe later in this meeting about how you really do that honest and transparent collaboration. And I'll stop there.
Okay, thank you, Lynn. So we'd like to open it up to the rest of the panelists. That includes the speakers. And I'd like to go back to Leslie. And we know that collaborations between researchers or bench researchers and clinical researchers are essential to facilitating drug development. Could you talk about the process of establishing these collaborations and what challenges might be faced during the process of translating benchtop research into clinical trials?
Yeah, I mean, it's an excellent point because in general, basic scientists and clinical researchers or clinicians, they speak a different language from one another, and that is challenging. But we do things in several ways. We have every 2 years, we have an international scientific workshop, and we purposefully weave in both the clinical science and the basic science. And so we kind of sequester people for 2.5 days, and they got to learn how to talk to one another. And in a field as small as progeria, you see repeats, which is great because people continue those relationships between meetings. But you're absolutely right. And the other thing we do is we try to sort of pull in basic scientists to some of our group clinical meetings, because actually the work done by Jay Humphrey that I showed was a result of discussions with him about what we're doing in the clinic and what the meaning of that is. His primary research is in vasculature, but not progeria. But once he sort of got the bug as to what is really important to us in the clinic, he was able to translate that into the mouse model. And so really it's just continued communication in any way that you can and pulling these people together, even though it's difficult because they're big, they're not trained the same way, but they can crosstalk.
If I could add, David, to what Leslie
Yeah,
just
please.
said, I hear you, Leslie, completely. And I would also add that I feel like maybe regulators speak a different language than clinicians and basic science researchers. And drug companies speak a different language even still from there. So, I do feel like there's a need to kind of understand how we all work together, because particularly in these fields, as we're talking about translational science and how we use surrogate or biomarker endpoints, it crosses all of those disciplines and we're all talking different languages.
Yeah, I completely agree. It's such a challenge, but at its base, everybody just wants this to work. And so I agree. I can tell you that for me, oh my gosh, going in to speak with either a drug company or the FDA is such a challenge. Different backgrounds. Everybody is incredibly well-meaning, but it's a challenge to get on the same page, but we do it. And the more contact we have with each other, the better.
That's great. Thank you very much, Leslie. Now, there were some other questions on a slide. I was wondering if we could pull that slide back up from. Or is it up? Oh, here we go. Okay, so we can't get through all of these questions, and we've addressed some of them already. I'd like to skip to the last question. And is there more that can be done to encourage use of the types of translational approaches that we've been discussing? And so let's see if there are any of our panelists that would like to. How about Estelle? Would you like to comment on this and in light of your experience?
Yes, I think there is definitely, as I could hear from all the people who just spoke, the need to share more and to be transparent. And I think what is also very valuable, and that is very rarely done, share failures, right? Share things that ultimately did not work, where actually wrong predictions were made and potentially impacted negatively a program, or even worse, patients. Something, this kind of data, I think, should be shared as well as our success stories, right? And I think we all have examples like that. And ultimately, I was focusing today on the use of new approach methods and use of in vitro approach for to enable entry into human. Here, of course, I think I would really encourage sharing all the data that were actually successfully regarded by health authorities around the world, but also then later on when you are in the clinic, sharing the clinical data and providing eventually the validation for the translational models used. I think we need these efforts to gain confidence in this approach, but I think also to eventually impact how these packages are being reviewed by regulators.
Okay, thank you very much, Estelle. I'd like to address the next question to Christine, and we actually have a more specific question that came in that's not one of the ones listed on the screen, and that is, Christine, if a digital biomarker like actigraphy is considered as a functional or clinical endpoint and not a surrogate endpoint, how do you demonstrate its clinical meaningfulness?
Yeah, that's exactly a little bit what we're struggling with, because when we look at actigraphy, are we looking at the absolute change in some score? There's lots of different markers that are coming out of actigraphy. So some of the approaches that we were thinking about was if we could maybe tie it to a PRO, so we know how much of change is on the actigraphy would relate to something that's beneficial that you can measure in a PRO. But right now, we still haven't really seen a good endpoint coming out of the actigraphy data. In children, it could be also kind of difficult to use, because as children grow and develop, then their baseline activity level is changing. So there's other challenges when trying to think about using actigraphy to support pediatrics, just because of differences in development. But if once we get a endpoint coming from the actigraphy data, that would be actigraphy itself is measuring a functional response. So to me, that would be more of a clinical endpoint. It would not be a surrogate marker, but we just have to figure out what is a meaningful change and exactly what's coming out of the actigraphy. What is the endpoint coming out of the actigraphy data?
Thank you.
I would like to
Yeah, go ahead.
back to like how about in terms of, how do we come together and share data? Yesterday I was on another workshop when we talked with CPATH. We were talking about ways to share data. And it is, we have to figure out a way of overcoming this challenge, especially with sharing of the clinical data. Because I was just thinking of the example that I presented for PAH. The reason we were able to use or develop PVR as a biomarker to be able to approve, right now it's the only drug for children, is because, the FDA has the data, and we were able to, in that case, invest resources to be able to make that link by showing the relationship between changes in PVR over a broad classes of drugs as individual drugs that are already approved in adults. But something like that would be very difficult to do outside of the FDA unless companies start sharing data. So what is the forum that allows companies to do that? I think that's just something we have to tackle, because I think there's so many barriers out there, because we're sitting in, companies are sitting in silos. They're not willing to share data. And then even if and I don't think having the FDA, even though we have all the data, I don't think that's a sustainable model for, being able to develop your biomarkers into endpoints or drug development tools. That's just we can do it on a limited scope, but that's not, a viable marker. So I think that is something I think just as the community at large, we really start needing to tackle, and how sharing of data can be done, and kind of trying to address some of the barriers to share data.
Yeah, thank you, Christine. I know there's been some successes with consortia, like Critical Path Institute or others, that in specific areas have been able to bring data together. Often it's placebo data. I guess, Tony, we'll circle back to you. I don't know if, you were commenting on this in the ultra rare diseases, it's people are more willing to play nicely together, I guess. And if you have any other thoughts on this, and also if you wanted to elaborate on your comment at the end before we went on to the rest of the panelists.
Well, I'll try to be brief, but I'll say a couple things, Dave. As far as the sharing is concerned, in my mind, and maybe I'm a little more cynical than a lot of people, but once a company makes a decision that they're interested in a certain indication or a certain drug, it is competitive. So as people said yesterday, it needs to happen in the pre-competitive arena, if you will. And that, to me, that seems to back up these kind of consortiums or relationships to patient advocacy groups and academics, really, to get the ball rolling, unless something else happens. So that's the conundrum for me, is that the resource people and are the ones that have to start things off and try to get the ball rolling. As far as the other concept, really, that I called intentional regulatory transparency, it's actually, I think it sounds really cool, but it's kind of simple and doable. And that is, for like a decade, I'm going to talk about NDA reviews. It's going to be scary here, but there was always this stimulus to streamline NDA reviews and not include regurgitative stuff that just was there because the company submitted it, and just to streamline them down and just say what you got to say here's why we approved it. Now, in doing that, that's great, and it makes it much more readable in a certain way. But I think the intentional transparency here is that to think back and say, what can I do to educate people or educate the public about how the data supported making this decision in a bigger sense? And like that might be including more tables than is actually 100% needed, or it might include that you need an appendix to put some information in about things. So that's what I meant in that sense by intentional transparency there. The other part about it is to go out of your way to let people see how you make the sausage. And that is, how is this data, how are we looking at this data, and what's the bar that had to meet to do this, to meet a biomarker qualification or a drug approval or something like that. They were the two things that I thought, just thinking about those when you're doing that. And, I know it's hard to do because you're really doing a lot of work when you're doing an NDA review, but just to kind of take a deep breath and say, here's the educational part, and here's the data part that you can put in those. That's it.
Thank you, Tony. I think those are some very good points. Wanted to circle back. Lynn, you have any final parting comments? We have just a couple minutes.
I do. Thanks, David. One of the things that I was thinking about that last question a lot, what we can do to encourage, because I think a lot of people who are listening want to know what we can do and what they can do. I feel like if you're listening to this, if you're at this meeting, I don't think you're necessarily one of the folks involved in drug development of the 10th Me Too drug, or, how do I make this drug in an oral solution because I want to extend patent life. A lot of people are listening on these calls are doing the hard work of trying to be innovative and get us unstuck in places where it's hard to get unstuck. And in that regard, I always feel like there's a sort of a tension between regulatory certainty, if you will, and innovation. Because regulatory certainty sort of implies you already know the path is well traveled, but innovation requires kind of taking some risks. And I think also part of what we need to do, I think it collectively, and whatever consortium or whatever sharing we do, that it's got to help all of the stakeholders, from the bench researchers to the regulators, kind of de-risk those situations, de-risk those decisions. And I think that the only way we do that, or one of the ways we do that, I should say, is to really think about, again, to Leslie's point at the beginning, who are we working for? What are we willing to accept? And then to have the honest conversations about that. And not necessarily, I don't know how you do that pre-competitive or competitive. Really, that part I'm not clear about. But I do think that in order to do that, we have to come to the table and we have to be willing to have that, basically honest conversation and try to figure out what is really risky for each of us, and then to talk about how we can de-risk it for each party. Thanks.
Okay, thank you, Lynn. Thank you, Tony. Thank you, Leslie, Estelle, and Christine for the excellent presentations and panel discussion here. We're now going to move on to session 5, our closing session, which will be moderated by Dr. Michael Pakanowski, who's Director of the Division of Translational and Precision Medicine in FDA's Office of Clinical Pharmacology. Mike?
Thank you, David. Excellent session, great discussion. Welcome to session 5. This is the last session of the workshop, and in this session, we're very fortunate to have some key leaders that really bring a very diverse set of expertise and experience from various sectors: regulatory, pharmaceutical industry, consortia, and research-focused advocacy organizations. And this panel will really focus on sharing perspectives on the state of the translational science. So for this session, we'll have no presentations. Our panelists will take some time to reflect on the key themes we've discussed throughout the course of the workshop. And I'm looking very much forward to this conversation. So panelists, I'll ask you now to turn on your videos, please. And I'll do a quick round of introductions of everyone. So we have Jen Farmer, who's the Chief Executive Officer for the Friedreich's Ataxia Research Alliance. We have David Reese, who's the Executive Vice President of Research and Development at Amgen. We have Eliza Thompson, who's the Deputy Director of the Division of Cardiology and Nephrology at the Center for Drug Evaluation and Research. And lastly, we have Steve Hoffman, who's Associate Vice President in Research Partnerships and Director of the Biomarkers Consortium at the Foundation for the National Institutes of Health. So in terms of objectives for this session, our esteemed panelists will discuss some of the opportunities to increase the use of translational research studies to support clinical development and achieve regulatory acceptance ultimately. Panelists will discuss how other programs may be able to learn from the successes and failures we've seen in other therapeutic areas, and we've certainly heard about several of those over the course of this workshop, and then how we go about best facilitating collaboration to achieve those goals for the drug development programs. I do want to remind everyone in the audience, you can feel free to submit questions for our panelists via the question and answer function at the bottom of your Zoom screen, and our speakers will be able to comment on them. So, to really kick things off, I think, the first question perhaps on the minds of many is, with your wisdom and experience in your various areas, after reflecting on all of the different discussions at the meeting so far, what do each of you see as the key strategies for optimizing the use of surrogate endpoints, biomarkers, or other translational approaches in the drug development context? And to kick us off, I'd like to hear the pharmaceutical industry perspective. So David, if you'd be so kind as to start off with a little bit of your reflections on the meeting so far.
Yeah, thanks, Michael. And I think it's been a great meeting. As I think about the discussions over the last couple of days, one thing that strikes me is certainly from my seat as the head of R&D for a biopharmaceutical firm is making a biomarker fit for purpose. And so starting with the simple question, what is the purpose of this biomarker? Is it something simply, as simple as a pharmacodynamic marker, a marker of target engagement very early in drug development or in preclinical development? Is it a surrogate for efficacy or safety? Is it used for patient stratification or selection, essentially a molecular definition of disease? And then finally, is it a marker for response or resistance, i.e., are we using it to guide our assessment of therapeutic outcome? Within those categories, I also think about, am I using this biomarker for decision-making purposes? If so, I want to have a certain level of validation and certainly a high degree of confidence that I can rely on changes in that biomarker to guide decisions that are often very high stakes. Am I ultimately going to be using this as part of regulatory approval or a regulatory filing package? And a subset of that question would be, does this biomarker need to grow up and become a companion diagnostic of some sort? So to me, establishing where it fits in that landscape up front is critical. And we often forget these sort of basic questions and the basic notion of what is the purpose of this biomarker as we get engrossed in the technical aspects of it.
Thank you for sharing that. I do have some follow-up questions, but we'll hold them for the time being, just based on a couple of things that you mentioned. Eliza, if you'd be so kind as to share your perspective, having been in regulatory review for over a decade at this point.
Yeah, and I think David made some excellent points, and in thinking about this question was maybe thinking about it from a little bit of a different perspective. I think that yesterday, we were reminded that it's been about 20 years since the Critical Path Initiative, FDA's Critical Path Initiative, was launched. And really, this initiative was intended to modernize, among other things, our toolkit, right? It was supposed to modernize how we develop medical products, but in particular, help us discover and develop a better tools to assess and predict and demonstrate efficacy and safety of products. With that in mind, I feel like we've been tremendously successful, and I think we should be very proud of the fact that we've developed, I think, the necessary infrastructure, whether it be the external infrastructure in terms of developing some of these public-private partnerships, or really the internal structure that we needed to develop to really support some of these discussions. I think we've also been tremendously successful in terms of really outlining key principles that need to be considered. I think just reflecting on some of the stories we've heard over the last few days, I think they speak to the importance of basic science and understanding disease mechanism, really in terms of developing biomarkers, how good it is or important it is to have good animal models, I think they also speak to the importance of data sharing. And I did just want to make one point that circles back, and then I'll stop, I promise, Mike, to what Christine ended on, because Christine and Mike as well, we were at this workshop over the last few days, which was a workshop related to safety biomarkers for drug-induced kidney injury, which we currently lack. But I think the critical piece that we lacked in terms of the data sharing, at least in that space, was really getting the patient voice involved. The patient voice is powerful, and I think moving forward, getting that voice in the discussion of data sharing is critical.
Excellent points. And I think that's a two-way street, I think, as well, with working with the patient communities and not only hearing from them, but also sharing the kind of things that we think about as part of the drug development process too, are very, very critical, just from an educational standpoint. With that wonderful segue, Jen, I was wondering perhaps if you could share a bit of your perspective.
Sure, thank you. It's been a great workshop, and really appreciated the opportunity to participate. I think a few takeaways from the last 2 days for me, and in some ways maybe some validation of things that have worked and not worked. It's been mentioned by lots of speakers what a long and resource intensive process this is, and just in terms of managing expectations through that process has been a real challenge, and I think continues to be for all stakeholders. And another challenge that I don't think we've hit on, but it's something that we've experienced, is thinking about all those different contexts of use within a disease that's a multi-system disease that's going to have multiple different treatment approaches. And so, where we've found ourselves on the strategy side of thinking about endpoints and biomarkers is really going back to what was in the first presentation yesterday, in terms of the chart of the different contexts of use, and blowing that out, and really forcing ourselves to prioritize which ones are the critical steps for drug development, and then really trying to drill down on those, because we can't have a biomarker for everything. It would be great if we could, but and because of the failure rate that we've heard about through some of the different case examples, we also know we're going to need to have multiple approaches for different contexts of use, and even trying to think about different therapies or different organ systems. So when you think about it in the context of a whole disease, it is a lot, and I think that's also what further drives the need for multi-stakeholder engagement.
That's an interesting point. It is often the case, I think, that we do, find ourselves with some technology in hand and trying to find a use for it rather than trying to solve the problem first and sort of beginning with the end in mind, so to speak. I think it is very important to be mindful of what our goals are, and certainly wherever there's been a critical drug development challenge, there's usually no shortage of people to rally around that issue and form those alliances to solve it. So that's an excellent point. Steve, you also work in the area of trying to facilitate collaborations and motivate and consortia. Can you comment a little bit from your perspective on some of the science and issues you've heard over the past couple of days?
Thanks, Mike. And it's always easy to go last and say yes, and I agree, and with everything that's been said. But I also have listened to a lot of the presentations, and I really appreciate the perspective that Jen and others have talked about staying focused on some of these pre-competitive efforts where you bring consortia together, where you're able in that environment to bring the right expertise and the right organizations together, the academic community, the pharma community, the patient advocacy voice, that all of those must be present really to have ultimate success, I think. And if you've got a missing piece there, you're going to struggle and it's going to take longer time. I've liked in, say, earlier today, Nicole's presentation where she went through with MRD, where you have MM and AML and ALL, that type of a phasic, or we kind of call it a franchise type of approach to different projects, works pretty well, where it may be two different ways that we do this together. You get all the right people at the table, and you might be using, let's say, MRD and technologies around that, and you do this for myeloma, you do this for AML, you do this for ALL, and you start to build this body of evidence that leads to, say, surrogacy and the ability to do that. And then at the same time, you get the shiny new tool that Jen was just talking about, and everybody goes, oh, let's throw spaghetti against the wall and see how this shiny new tool works. And I have 7 contexts of use that I would like to use it in. Let's go ask FDA what's the right one they care about. But when it's the other way around, it's getting that expertise and that focus to decide what is the more strategic need now as step one. And maybe you work on a diagnostic or a prognostic marker that goes through the qualification process, therefore begats adopted universal use. And then that then becomes a pharmacodynamic response marker that leads to greater treatment response and or trials that are using that biomarker, and it gets to surrogacy. So kind of managing those expectations, like Jen said, up front, and trying to get that path going forward is, I think, what Consortia can do perhaps a little bit more within Safe Harbor and sharing of data to get to that path. So that's kind of my recommendation for success. Obviously, our organization, CPATH, and others are very much built to try and do that.
And that's thank you for sharing. That's a tremendously valuable insight. So we have, I think, over the course of the meeting, heard a very broad spectrum of levels of validation, so to speak, and different types of experimental models. And, it's hard to talk about everything in this session, obviously. We can't address all issues and contexts of use and whatnot. But I would like to go back a little bit to David, if I may. And, you mentioned some things about, there's some information that's essential for you to understand at various decision points in drug development. The gravity of the decision, I think, is something that weighs, heavily on what you think about when looking at a biomarker. I'd like to ask if, as you're thinking about designing a drug development program, in the pharmaceutical industry, when looking at sort of the clinical trial assessments or even, non-clinical studies that would be performed, what kind of things do you think about would help improve utility of those kinds of information streams in your decision making? Are there certain things about the validation process, that make them more apt to be used in important decisions?
Yeah, I think that's a hard question to answer generically, but let me take it up one level and say, certainly the approach we're taking is predicated on a belief that we're entering what we like to call the era of human data, from in silico prediction of protein structures and protein-protein interactions to multi-omic profiling. I'll come back to that in a moment, to enriched clinical trials data. And then there was a question that just came in about real-world evidence, real-world data. Of course, there's a tsunami of real-world evidence and real-world data now washing over it. And so it's my real belief that it's the integration of all of those data sets that's actually going to propel drug discovery and drug development. And many of them are actually related to biomarkers. Now, they may well be multi-parameter biomarkers. And in fact, as we evaluate a program, starting with a target and a pathway and validation, we now bring multi-omic profiling to bear. A decade ago, we acquired a company called Decode Genetics, one of the best genetics organizations in the world. I was just in Iceland last week with others for the 25th anniversary of Decode, but it's not just a genetics company now. In fact, when we acquired them, they had done a couple thousand whole genomes, had, tens of thousands of individuals that had been genotyped. Now there are hundreds of thousands of whole genomes, with 15 to 20,000 being added on a monthly basis, and many, many tens of thousands of individuals where we've got transcriptomic and proteomic profiling. So it's actually the integration of those data sets and then ultimately reducing them into practice for decision making that I think will propel our fields. And I think for some diseases, it's in fact almost the only way forward. I'll just end with one example, a disease like lupus, where, right now we're at the level of clinical phenomenology in describing the disease. And in fact, the diagnosis is based on having 4 out of 11 things on an ACR checklist, but that doesn't tell us a lot about the pathogenesis or what we ought to do. So, and we know that some patients have kidney predominant disease, some it affects predominantly the skin and joints, et cetera. We know there are molecular subtypes hiding there. We just don't know what they are. So to me, the real power coming forward is going to be to answer those sorts of questions, to then come to a much more intelligent starting place in the drug development and discovery process.
It's interesting, you sort of mentioned the use of sort of these multi-omic approaches, and I was just wondering, has that required a cultural shift, not necessarily within your company, but do you think there's been a cultural shift that's more inclined to look at those types of analyses than perhaps in years past, where it might have been more prudent to just kind of collect only the essential data for a drug development program and not be distracted?
Yeah, I think it's coming. There's also a danger, because with these sorts of complex analyses, the one thing you will find are a lot of correlations. And the one thing you want to know is which one, what tiny subset of them are actually true. And so, there's tremendous promise, but a cautionary note as well, which is that the analysis, the analytical engine, ultimately is the rate limiting step here.
That's interesting, and it also, as was mentioned, we were in a workshop on drug-induced kidney injury over the past couple of days, and that sort of issue comes up with a lot of transitional biomarkers, that they can be very potentially distracting. You don't want to be misled by information that's not well validated, and perhaps maybe I can turn to Eliza, just maybe with that kind of issue in mind. What do you see when we're looking at data from either a novel biomarker or a new model? What do you see as sort of the regulatory view on those kinds of things, and what are you finding as the main challenges in that sort of discovery to validation life cycle?
Well, I think, as all have noted, it's really a heavy lift, and unfortunately, it is a long and resource-intensive process, which again gets to this, whole issue of it takes a village, and we need to work together. I think, at the basic level, you have issues, as others have noted, just even with analytical validation, and then there's even understanding things like biologic variation or variation in other populations, or a variation due to demographic factors, and all of this on top of then the need to really have access to the data sources and data that allow you to do the clinical validation piece. So it really takes a village, for lack of a better way of saying it. I think the other things that have come out in some of the other talks as relates to this is, in some of these disease spaces, our gold standards aren't great. And in one space in particular, and this harkens back to our workshop on drug-induced kidney injury, is, we're trying to figure out a biomarker of injury to the kidney. Unfortunately, the only thing we can relate it to for often is actually changes in kidney function, which may not reflect injury. And so, that makes it even more, challenging when you're in a space like that. I think the other challenge we have, and this is true in a lot of our, certainly I think some of our rare diseases and sometimes our common ones, which is, as the science advances, we're discovering either new diseases, or really we're sub-segmenting our population of a disease into other more distinct subsets. And then when you try to sort of figure out the natural history of the disease or use the data that have been collected up to date, it becomes more challenging because, 10, 15 years ago, no one was really calling at this. No one was really measuring this that we now measure to subsegment the disease. So, a lot of challenges, but at the same time, we've had a number of successes. So I think there's reason to be hopeful.
And definitely something, as maybe Tony Dermowitz alluded to a little bit before, the landscape of cystic fibrosis has evolved over time. And, that by necessity has, required us to think about different models and such, sort of along, the same lines of, we often talk about qualification and, there's a very high bar for that. And even qualification toward the goal of surrogacy, whether for Subpart H or traditional approvals. Do you find that there's a difference in the conversation with individual drug developers and how we think about biomarkers for a specific program and a specific medical product versus the whole universe of kidney disease, for example, or chronic kidney disease or kidney injury? Does that influence your thinking on what's required?
No, I think it's just the level of evidence, frankly. This is, I think as others have noted, there's really a number of ways that, these biomarkers, enter our toolbox for use in drug development programs and regulatory decision making. And it's not so much the way in which they enter it, scientific consensus or use in an individual drug program or qualification. Ultimately, it comes down to the evidence that supports its use for that purpose.
Mm-hmm. Yeah, so Eliza mentioned, it takes a village. We've heard that, I think, several times over the course of this meeting. It certainly takes a lot of parties. And, Jen and Steve, I think you both have worked in trying to set up efforts where you're either trying to accumulate patient data or trying to get companies to collaborate. And it was alluded to a little bit earlier, that there is an issue around, a single company's motivation or incentive to contribute to those kinds of efforts. It can be very arduous and expensive, as has been noted. And it may not always necessarily benefit, one given company. And, obviously in rare diseases, there may be a greater need and pressure to develop these consortia and because there's a lack of medical therapies. But when, in your experience in sort of starting, grassroots efforts or even now working at more advanced stages of consortia activities, what have you seen as sort of the key strategies for facilitating interactions between stakeholders, whether they're, academics and pharma or patient groups or whatnot? And Jen, maybe we'll start with you.
Yep.
I think of a few key things when I think about some of the collaborations or consortia that have worked or are working. One key piece, as others have mentioned, is just the role that the advocacy community can do just in bringing people together, because one-on-one, everybody kind of tends to, everybody meaning different stakeholders will think that their issues are unique or, specific to them, or that there isn't pre-competitive space. But if you can really bring people together, and it's really helpful. I've leveraged the academic community a little bit to be able to, initiate these kinds of dialogue to identify what are the common issues that are not competitive, or if the academic community starts to really talk about how they want to, in the case of biomarkers or something like that, directions that they want to go or ideas that they have, if our industry partners and our regulatory partners are also in the same room, I find that more times than not, it's harder for folks to not participate than to participate. And that just you catalyze this collaboration that's needed, and you start to gain some common ground, and it's an organic kind of thing, but if you can really promote the initiation of that, it has the catalyst to be successful, and helps people realize that they really do have more in common in the ability to work together, and still have the ability to achieve success, or the individuality that they need, that for the academic community, what they need to get out of the collaboration is different than what our industry partners need to get out of the collaboration. And so there is just the ability to help people realize that, and to kind of be the negotiator and the translator in that process. If you have an entity that can really do that, then I think you can be successful in these multi-stakeholder consortia.
I'm going to continue to use Jen as my setup, which works really well for me, and I appreciate it. But I also think, now David talked about novel technologies and this molecular characterization now in the technology world for biomarkers that's ongoing. So in some ways, we're stepping back to step forward, where we almost have to go backwards a little bit, come up with all these new molecular characterizations of diseases. These may be diagnostic or prognostic markers to then be able to move forward with the regulatory efforts. And I do see the consortia can bring together a lot of novel tools, technologies, analytic approaches, and a lot of times, and I'll use our accelerated medicines partnership kind of partnerships that many folks may know about, where there's these data portals and these knowledge portals, and they also have novel visualization tools, analytical tools that can be shared across multiple partners that might be able to analyze their own data with those same type of tools, as well as the data that's within a project that moves forward. So there's some core benefit to multi-stakeholders that are involved in that, getting access to some of the raw data within the partnership early on in an accelerated manner. Companies don't want to publish, and they're not too worried about those types of publications. If you can accelerate those efforts to benefit the academic community, have the communication piece that gets out to the patient community, while at the same time allowing both researchers and industry to interrogate that data at their own speed, wants, needs, as well as together, you really can accelerate things in that way. And I think that's where the consortium approach can be a real benefit to the field.
And you've obviously worked over the years in aligning sort of many stakeholders, and if you had the ability to make a request or a plea of your counterparts in pharma or patient organizations or pharmaceutical or diagnostic companies rather, what would it be? Do you still see cultural issues that need to be overcome, or are we on the right track?
I'll start on two things. I certainly think there has to be particular willingness to share data in a safe harbor way. We can do third-party analytics. We can do things in a way that masks and conglomerates things and blinds things. So there needs to be continued openness of companies to share data, and that includes negative data, right? Negative data can be extremely helpful and beneficial that could be put into this. I'll actually, Mike, talk to you guys as well, right? When you get to the point, particularly through the biomarker qualification part, where you guys have approved LOIs, you have approved qualification plans, I feel like you could be even more strongly involved in what that analytical approach is, maybe even bringing some insights from data that you have internally to what's the right way to do that. And we don't want to yap about the time that takes in there. It's obviously a difficult and time-intensive effort, but turning that around, because the project's continuing. So we need that feedback from FDA, where your thinking is. And if we're to the point of a qualification plan that's approved, why are we not doing that almost 100% together? Because it's not about a particular product, it's about that biomarker to make a regulatory drug decision. Can we do that even more in unison? How do we do that? Maybe a consortia environment's a little bit better and okay for FDA than perhaps helping compare a diagnostic with an individual company. So maybe there's a little bit more, I guess bias is the best word to use it, for those consortium-based efforts for biomarker qualification, as opposed to a companion diagnostic with a singular company and drug.
David or Eliza, did you want to react to that? What Steve says resonate with you, or?
Yeah, I mean, to me, and yeah, I think it's quite accurate. I think the data sharing piece is often, and the reluctance around it is often more theoretical than real. And it's actually a surrogate itself when there's discomfort about what are the actual questions that are going to be addressed by the consortium. What is the scope? What are roles and responsibilities? At least in my experience, and we've participated in many of the FNIH consortium, for example. Once you address those questions, a lot of the data aspects will actually fall into line. Now, sometimes there are sensitivities, but it's not, at least in my experience, as rate limiting as conventional wisdom might suggest.
And maybe I will just offer quickly. So Steve, I wish that Jeff were here to answer your question as the person who heads up our Office of Drug Evaluation and Science. But I do want to say that, at least as someone in a review division who, we rely on, biomarkers for everything we do. I do wonder and often think about what more we could be doing as a regulatory agency to support this, either using internal data or, as you note, really having an additional layer of involvement, or just the speed with which we get back to you. And I think some of this is frankly just a resource issue. But again, I can't speak for Jeff, who knows a whole lot and has thought a whole lot more about this issue.
And Eliza's been great on the kidney side, so don't let me think that.
So I think this is very helpful. It's good to hear that there is, because sometimes the reluctance to participate in consortia is you can't hear if it's myth or if it's reality. But Jen, from your perspective, as you sort of look at all the work you've done over the years and other organizations that have been successful at kind of building a scientific infrastructure like Cystic Fibrosis Foundation, for example. What would you say to folks that are in a more formative stage, who are just beginning to develop natural history studies, for example? What comes first? What are the resource needs? What was your biggest lesson that you've learned over the course of the years?
Maybe, but before I answer that question, I just wanted to come back to the challenge question again, because I think from my recent experience, the challenge is actually, I do see more progress on the data sharing side. And I think that is a hurdle that we're overcoming more quickly. The one that is really challenging is the legal. What holds up development of these consortia and participation is legal. And I don't have good suggestions for how to fix that, but and it's not a industry or academia specific issue. It's across stakeholders where the legal issues really are challenging, especially when we get to multinational consortia. And I spent 18 months in legal negotiations just to get a consortia off the ground, and before they could do any work. And several members had to drop out because they just we couldn't get there for them. So I feel like that's a place where those are problems that should be solvable, it feels to me. But coming back to kind of, if you're just getting started and thinking about putting in place some of these resources, don't underestimate, as I think Leslie gave a great example of the power of your ability to bring people together at conferences or meetings, and the power of also thinking about the all the different stakeholders in the same room, and how that learning and sharing really can accelerate and translate both the basic science as well as clinical development. And the one piece that's been really humbling to me over time, and that I think organizations really need to think about, is sustainability. I naively thought when we started in 2003 with a natural history study that 5 or 6 years down the road, we'd have the data that we'd need and we'd be done with the natural history study. And it's 2022 and we are still doing our natural history study. And it's an evolving, iterative process. Not to say we don't have tools today that we didn't have back then, we do, but you're going to want to make them better. You're going to want to, make your drug development process even more streamlined based on those learnings. And so you have to think about how putting something in place can really be sustainable over a long period of time. Because I think somebody else mentioned it earlier today, it's really the advocacy groups that are there for that long period of time.
And I think, one takeaway from your comments is perhaps we'll invite some lawyers to the next meeting and have them at the table as well, because that's sort of the next dimension, I suppose. So as we begin to sort of think about what next steps look like, and sort of what the future is, and increasing acceptability of different approaches as we sort of wind down with the last question for the panel discussion here. I was going to ask that you all try to do it in 10 words or less, and you can feel free to do so. But we have a few extra minutes, so if you care to take some time and think about or propose what you think might be the agenda moving forward, and what the gaps are, as sort of the globally and advancing science. Feel free to offer those. And since I've put Steve at the end every time so far, I'll ask him first this time.
Thanks, Mike. I appreciate it. It's easy to say. I think that having a consortia or bringing multi-stakeholder partners together, finding that pre-competitive space to answer these questions, actually having the clarity in the questions of really what you want to do, is I think is a great path forward. In the future, I think that we're often waiting for someone else to champion something through. And is there going to be this great academic with this cool idea that we want to support, create a project, and ultimately has the idea there? But I would even challenge to the industry, or for that matter, even the FDA, as the source of where we need to make differences, and then come to organizations like ours to rally around this. We did this very successfully back in the day on CAP and ABSI and HAP and VAP, where it was really driven through FDA needing some symptomatic endpoints to be augmenting those guidances and for drug development. So that was a very successful effort. And so often, instead of waiting for this, key leading academic in some autoimmune condition to bring something forward, and we nurture this 10-year project to get to the end, is there something at the ready, at the need, at the FDA level, or a company that is really kind of right at the preface of having something that they could really, in the pre-competitive space, get in other data sets, get in other technologies to move something quickly that's really needed in the field. There was a question in the chat related to what has COVID taught us. Clarity, need, absolute alignment, financial resources, willingness to get to the answer in 1 year and 2 years to this. Those types of things I think are very possible. Perhaps if we turn the turn it around a little bit, and it comes from, let's identify together where that high need is, bring it to these types of organizations like ourselves, GENS, CPATH, and others, and I think we could help spin that together very quickly. That's what I think the future could be benefited by. Thanks, Mike.
And just to ask one question, that's very, very helpful. When you think of the concept of safe harbor, where do you think that lives? Is that something that exists within under the auspices of FNIH and the Consortia, CPATH, and those, or something else?
I think we enable it. I don't know that it has to live under us. I think we can also bring in other organizations that are not so, I guess, individually vested, whether that's in data sharing and data standards and trying to come up with these types of portals. I think they could live wherever they need to live. But I think as a mechanism of that open sharing, of that privileged access type of a scenario, I think we can facilitate that, and that should be our role. It's not us. I don't need to be that owner or those types of things, but I think I could facilitate that in a group design and let that group think, figure out what's the appropriate level of sharing, appropriate level of how you can interact and access for those things. At least that's what's been successful for our organization.
Thank you. Eliza, I'll ask the same question of you. In as many words as you wish, what do we need to do next?
Yeah, and to be honest, a lot of great things have been said, and I'm going to maybe keep mine to 10 words. Actually, I didn't count how many words they are, but I think it is this idea of working together and sharing data. And I think if we do that, we're going to go farther and get there faster. And I do think that workshops like this are fabulous, as a way to get everyone together, to brainstorm, to share examples of success, and to think about what more needs to be done moving forward. So thank you for including me.
David?
Yeah, so to me, I think it's time to start thinking about the reduction to clinical practice as we enter a world where we're going to have much more complex biomarkers. There's going to be an immense educational lift for primary care physicians, whom in 10, 20 years are going to be expected to use these markers. And so that translation into ultimately benefiting the patient in the bedside, in the community, to me is something that we have to start thinking about now.
And do you think, your company has experience in, cancer drug development, obviously, and companion diagnostics, and, a lot of experience learned from that. Is there any wisdom in particular?
Yeah, I think there are probably a lot of lessons from oncology where this, because of the state of the science and the state of drug development has really penetrated first. But even there, we're not perfect. Some biomarkers, we still only have 80 or 90% of the patients who should be tested for that biomarker, the tumor tested for it, are actually being tested. So you're right, a lot of lessons there that will become even more complicated as we move into different disease states.
Great, thank you. Jen, we'll let you have the last words.
Thanks. And maybe I'll shift focus a little bit to just say, what am I optimistic about after the workshop? And one takeaway for me was that, when you have good treatments and or lots of trial data, you really can accelerate this process. That was, a really hopeful and encouraging takeaway from the case examples I heard the last 2 days that, get me excited to go back to our team here and think about, how we could be doing things maybe a little bit differently, or, how the time we've already put in and the work that we've already put in will get leveraged as good treatments come forward. So that was one of the exciting things for me the last few days.
So I do appreciate ending on the note of optimism. Thank you very much for that. So that brings session 5 to an end. I really do appreciate all of your input. You've had a lot of experience in this area, and your advice is sage. So I do appreciate you sharing your thoughts in this session. So that brings this session to a close. You can feel free to hop off video and we'll transition to wrapping up the workshop. So, you know, I'd like to take a few minutes maybe to, you know, just provide some reflections on sort of the high points of the meeting. You know, biomarkers and translational tools have for a very long time been a major focus of FDA and a lot of our research efforts over at least the past 10 or so years that I've been at FDA. It was about 10 years ago now that we had a workshop on advancing the use of pharmacogenomics and biomarkers under PDUFA 5. And you know, the interest and intensity in trying to find ways to accelerate drug development and advance new technologies, I think has really only intensified in that time since. And I do think we all share that common interest of, you know, making the best informed drug development decisions and approval decisions. And there's no shortage of them. There's many different things that have to be addressed over the course of, you know, going from first in human even to post-market utilization. So the types of conversations that we've had today are critically important. And I think we've heard about, you know, a tremendous number of very instructive cases that I think will give us a lot of food for thought. I will say there's really, you know, a large handful of approvals in very recent times, past year or 2 years, that you know, where experimental models or biomarkers really played a pivotal role, whether in terms of supporting the substantial evidence or informing approval decisions, or which patients should get the drug or at what dose. You know, so this means to me, you know, that we are in fact making progress. And I think, you know, to watch this continue to develop over the years has been extremely gratifying. And of course, there's certainly quite a lot of work to be done. You know, so in thinking about, you know, the content of the sessions over the past 2 days, you know, we've heard a lot of, or today at least specifically, we heard a lot of echoes from the points yesterday about how biomarker development and gaining acceptance as a surrogate endpoint is really a long, resource-intensive process that indeed takes a village. And we also continue to hear about the importance of partnerships and ongoing iterative dialogue with the stakeholders. And our big themes today, I think that came out, a lot of the discussions were related to data sharing and standardization. And data sharing, I think, is really pivotal. It's clinical data, not only the positive, successful studies that have been performed, but also the negative ones. And past data, including, you know, failed studies, can help inform validation efforts and be applied to future studies. Now, I know data sharing can obviously have its difficulties, as we've heard, you know, many legal concerns and other issues, you know, but, you know, there are various ongoing international and national efforts really to encourage data sharing, especially in the areas of rare diseases. So there have been a number of successes. It is possible to do. And, you know, I think even as we heard in the last session, there's a willingness to contribute to it. You know, sharing in Europe has also been difficult for different reasons due to regulations, but there are efforts to standardize interpretation. So sharing can be happening with organizations within the EU as well. In terms of you know, standardization specifically, you know, having standardized data collection allows studies to be aggregated. It seems to be sort of a first principle, and the ability to aggregate data does lead to more opportunities for data analysis. And we'll always need to consider issues related to missing data and other, you know, factors that can make some of the information inconsistent, but it helps mitigate this when there are data standards available, and those can be adopted in putting together study information from disparate sources. In session 3, we heard 4 examples that came from a variety of different fields, but all sort of speaking broadly to the issue of, you know, use of biomarkers as surrogate endpoints and how they can be applied in research and clinical trials. An existing performance test of mobility problems in elderly patients was modified, you know, for children with hypophosphatasia. We had CSF biomarkers for neurodegenerative disease with different models to apply those as screening or enrichment or diagnostic tools. We've heard about surrogate endpoints for chronic kidney disease and how to best apply these endpoints for trial populations and treatment. And we also heard some case studies in oncology related to pathologic complete response and the regulatory considerations around biomarker development, including the various potential uses and validation and evidentiary criteria. In the last session, or in session 4 rather, we heard 3 presentations, again, about different translational approaches. We had a mouse model that was informing drug development and treatment in progeria, which is an ultra rare disease. We had in vitro models and ex vivo models that were informing the therapeutic index and risk identification in Wilms tumor and acute myeloid leukemia. And then we heard some information about the use of bridging biomarkers to extrapolate efficacy from adults to children in the setting of pulmonary artery hypertension. So, I think we've had a really tremendous tour of a variety of efforts happening in, the translational science space. We closed with some comments from some key opinion leaders in the field, and very much take their feedback to heart and kind of see what they view as the next steps forward. So, that's it for summarizing, most of the sessions today. In his opening remarks today, Mark, said that success for this workshop would be an open and lively discussion. I think it's fair to say that we achieved that. So I just want to, in closing, say I thank you all for your attendance and devoting your time, to this workshop. I hope each of you found the discussion fruitful and it sparked some interest in trying to move the needle forward and move the needle within your organizations. And I think it certainly, speaking from a regulatory perspective, has given FDA much to think about. This workshop wouldn't have come into being were it not for the speakers and panelists, so do appreciate that. I do extend my sincere thanks for your contributions yesterday and today. And lastly, of course, I must recognize the leadership and diligent work of folks that were heavily involved in the planning, specifically York Tamita, Megan Echelsani, and of course, Jeff Siegel in conceiving and designing this workshop, and really working hard to shepherd the FDA team through the planning process. And also many thanks to Aaron Sewell, Ethan Chupp, Mark McClellan, and the team at Duke for facilitating what I view as an excellent meeting. So that brings us to a close. I do look forward to seeing you all again soon. I hope you have a great evening and take care.